of motor-related twitches. Although we have learned last week that twitching is actually really important for your brain. So it is really great that you're here, Narender. We're really looking forward to your talk. Right now you're in London. Okay, what we're going to hear then is about the cerebellum, but it's very much this perspective on cerebellum where we start to think about it as part of the broader system. I think this is really important. All right, so pay close attention. Thank you so much for joining us.
Thank you. I just want to also say thank you for the invitation, and it's a fantastic lineup of speakers that you have here. I like to sort of quote Chris Mile, who was one of my previous mentors, about the cerebellum. He jokingly often used to say, well, the cerebellum is just the cushion that the rest of the brain sits on. But anyway, the talk is going to focus on the following things. These are the themes that I'll pick up in various ways. I'll focus on the cerebellum as a neuronal learning machine and perhaps try to rescue the reputation of the late John Eccles, who wrote a book called with Masao. That's right. The cerebellum as a learning machine, one of the best primers on the beautiful anatomy and physiology of the cerebellum that's ever been written, and I don't think will be surpassed actually. But I have to say I do agree with you about some of the other slightly odder things that Sir John wrote in relation to consciousness. But moving on from that, we'll talk about the anatomical organization of the system. Dick Passingham, from whom I learnt a lot as a postdoc, used to say that everything starts with the anatomy. If you want to understand a system, you need to start with the wiring diagram. And so we asked the question about a very controversial topic, which is, can cerebellar wiring actually support the idea that the cerebellum can be engaged in the processing of signals from places like the prefrontal cortex and participate in the process of higher cognition? So we look at the evidence for that, and we look at some of the pros and cons, and we also confront some of the objections that have been raised by slightly more conservative thinkers in the field. We'll then talk about the important topic of evolution. So what's special about prefrontal cerebellar connections in humans? Is there evidence for selective evolutionary expansion of the connections between the prefrontal cortex and the cerebellum? And we'll then get lost a little bit in theory. We'll talk about the way in which conventional ideas about human cognition, dual process theory, and so on, can be impacted upon by this kind of data. And I'll dabble a little bit in control theory, and I'm aware that I'm somewhat like Daniel in the lion's den here, because there are lots of people here who work on control theory who will shoot the ideas down, and that's fine. We'll talk about the ways in which control theory not only applies to motor control, but might also be extended into the domain of cognition. And I'll then talk about some fMRI studies that people in my lab have done on cerebellar plasticity and its role in supporting cognitive skills. And we can speculate on what this loose term cognitive might mean and so on. But essentially, you'll see that we've used instrumental learning as a behavioral model to investigate this. And that sort of behavioral model allows us to bring the process of cognition very much under experimental control. And so we investigate the automation of rule learning. In doing so, another theme that I'll pick up on is classical conditioning. And this is where the roots of my training begin. And since this is a summer school, I'll just mention that I picked up my training in the lab of Chris Yeo. So my career started as a PhD student at UCL, where I spent 3 years teaching rabbits how to blink. But that's fine, I profited from that, and I investigated that using, investigated cerebellar involvement in that. The person who was next door to us in the lab was Mitch Glickstein at the time, and Mitch and I have gone on to have some, shall we say, friendly disagreements about the role of the cerebellum in cognition since then. Actually, some of our interactions have been less friendly, but that's fine. So that's basically the themes that I'll pick up on. And we'll begin with what the traditional view is of the cerebellum and movement. We've known for a very long time that cerebellar lesions fundamentally cause motor deficits. They're the most obvious and the most well-documented symptoms that arise from a lesion. Gordon Holmes in 1917 wanted to get a better handle on whether this was just movement in general or whether this is something a little more specific, and by looking at patients, he actually gunshot wound patients from World War I, he quickly realized that actually the symptoms seem to cause a decomposition of movements. The seamless flow of a movement that uses several muscle groups seems to break down. These movements decompose into their separate elements. And what one realizes after looking at this is that actually coordinated movement, the process of coordination, is acquired, it's learned. And maybe what the cerebellum is doing is learning to stitch these various movements together. So movement becomes effortful, and patients frequently report having to attend more in order to execute voluntary actions. One of the things that emerges from our understanding of the cerebellum is that it allows us to execute these movements without the application of attention. And fundamentally, if motor coordination is learned, then the plasticity for motor memory, perhaps that's stored in cerebellar circuitry. And in some ways, another part of the story begins here with the work of David Marr in 1969, who's in some ways better known for his work on vision, but many people don't realize that actually he made an instrumental contribution to our understanding of how the cerebellum might work. And his inspiration was the local circuitry of the cerebellum. His inspiration was to look at the beautiful primary neuron in the cerebellum, the principal computational unit, the Purkinje cell, and to understand that each Purkinje cell interfaces with several parallel fibers that bring into it the context, the contextual information from many, many different parts of the nervous system. And in contrast to that, we have the climbing fiber. This is a single neuron located in the brainstem, in the inferior olive, that sends a single projection that has multiple interfaces with any given Purkinje cell. And that's said to convey an error signal to the system. An error signal for what, you might ask? This is basically to teach the system which synapses it forms with these parallel fibers are correct and which ones are not. Okay? So, along with James Albus in 1971, he produced they didn't publish together, of course, but Albus drew very heavily from the work of Marr. And both of them produced highly regarded models of cerebellar learning. And one of the interesting things about these models is that not much has changed over the years since then. You see, these are very robust models. So the idea is that motor memory could be stored in the connections, in the the plastic connections that are formed with its inputs. And just a quote from those two authors in 1971, one of the interesting things that James Albers pointed out that seems to have been borne out repeatedly in the experimental literature is that synaptic strength will tend to zero asymptotically. So the synaptic strength decreases from trial to trial. This is one of the messages he puts forward in his computational model. And there's a big literature now that suggests that, yes, this is probably what happens. And David Marr also suggested that high-level commands from the cortex, when he says cortex, he means the neocortex, could access low-level cerebellar representations, so that when movements have been learned, a simple or incomplete message from the cerebrum will suffice to provoke their execution. The idea being that you have a very high-level command in the neocortex that triggers the execution of processes that result in quite refined and sophisticated information processing that kicks off from the cerebellar cortex. And all of that applies to motor control. But in recent years, it's been suggested that this could be not just a computation that applies to motor control, but a universal transform that could apply to a whole range of other forms of information processing. And maybe there's scope to think about the cerebellum having a role in other kinds of skill. Maybe there are high-level commands from other parts of the neocortex that take advantage of those sorts of operations. You should feel free to stop me and ask questions, by the way. So we go back to the beginning. We go back to a very simple model of motor learning that's been widely used to understand the way in which the anatomy and the physiology of the cerebellum can contribute to learning. How is it that that learned representations can be stored in that circuit? And so this is how classical eye blink conditioning works. You have an unconditioned stimulus, which is an air puff to the eye. Of course, when you receive an air puff to the eye, that causes you to blink, and that blink is the unconditioned response, and that's typically characterized here. So you have a US pulse, let's say that's about 50 milliseconds, that provokes an unconditioned response, a closure of the eyelid. And in any classical conditioning model, you have a conditioned stimulus, and very often this is an auditory tone. So you present the animal with, let's say, a 300 millisecond auditory tone that co-terminates with a US. Of course, in this example here, there is no US, and that's simply there to illustrate the fact that it really is a conditioned stimulus. Before learning, it produces no response. Okay? There's no reason, unless the auditory tone is very loud, there's no reason why this rather arbitrary stimulus would produce an eye blink. But when they're presented together and you repeatedly pair the presentations, you start to see the emergence of an anticipatory response. And from trial to trial, the magnitude of that response gets bigger and bigger until it reaches full form, and it tends to peak round about the time of the onset of the US. So it's a remarkably well-timed response. And it's learned so well that even if you omit the US, let's say every 10th trial, in the case of rabbit eye blink conditioning, you still get the CR, of course. And one of the big advantages of presenting information in that way, of omitting the US, is that you can understand the full form of the CR without any interruption from residual eye blink reflex that is actually caused by the US. And when you do that, you see that if you plot every 10th trial, there's an emergence of a conditioned response, even in the absence of a US. And it's very well timed because it seems to peak at round about the time that the US would normally be expected. So what's all that got to do with the cerebellum? A number of labs, including the lab of Chris Yeo, where I did my PhD many years ago now, have characterized the responses of Purkinje cells in a very specific part of the cerebellum, that's the anatomy of which is understood now in exquisite detail. So the anatomical pathways for the US, the UR, the CS, and the CR are very well understood. The eye blink microzones have been very well identified. And if you look here, I apologize, actually maybe if you dim the lights. If you dim the lights, you can probably see this a bit better. Okay, doesn't matter. They're thinking. That's fine.
No, you want the lights dimmed, we get the lights dimmed.
Okay.
Less light.
Thank you.
Blink the light.
Okay. I'm training myself not to blink at the moment under this light, but anyway. So you have a tactile US, which arrives into the trigeminal nuclei and that synapses with facial and oculomotor nuclei, and you get the seventh cranial nerve that goes off towards orbicularis oculi in the eye blink musculature, and that simply causes a blink. So this is the US and UR pathway. Okay, so on top of that, you have another set of pathways. So you can ask the question, well, first of all, if the cerebellum is the target, how does the US information reach the cerebellum? The answer to that is that the trigeminal nerve projects to inferior olive. The inferior olive then sends climbing fibers to lobule H6, Purkinje cells in the cerebellar cortex. But of course, for this idea to work, you have to have a convergence of the CS and the US information. And so auditory information, the CS information, arrives via a number of different brainstem relay nuclei to the pons, pontine nuclei, and the pons sends its parallel fibers off to the cerebellar cortex, to the very same Purkinje cells. So the idea is that you have plasticity at this synapse here, the synapse between the parallel fibers conveying the CS and the Purkinje cells. And when the Purkinje cells learn, what tends to happen is that the conditioned response that's generated as a result is transmitted to the eye blink musculature via the red nucleus and the facial nuclei. Okay, so don't worry if you don't pick all of that information up. I think the key thing to remember here is that there's a convergence zone in the cerebellar cortex where the CS and the US come together, and the output, the conditioned response output, is generated by Purkinje cells and is sent on. So that's the system that people have looked at for a number of years, and being either very stupid or very brave, when I left Chris Yeo's lab and went to start my I started dabbling in imaging at Queen Square at the Institute of Neurology with Dick Passingham. The first thing I said to Dick Passingham was, "Are you going to let me do eye blink conditioning in the magnet?" And this was back in 1998 or sometime back then, when things like this were very difficult. And he said, "You're mad, but I'm going to let you do it." And so I did a study where we actually measured the eyelid movement itself in the magnet, and I spent months in the workshop physically putting together the kit for this to work. But the upshot was that we conducted a study where we replicated the effects that people were finding in lower mammals in humans. So we found activity in lobule H6 that was time-locked very specifically to the CS. And it's important to remember that because these are trials in which there was no US present, but there was a CR. Okay? We showed presence of learning-related plasticity in the human brain for the first time using classical eye-blink conditioning. What we also found is a similar profile of plasticity in the face areas of motor cortex. And these areas are connected. If I take a step back here, you'll see that primary circuit that I described, a system that people have not looked into very much, and one that I'm starting to look into now, is the way in which the motor cortex starts to influence these areas. The way in which the motor cortex begins to exert instrumental control over these conditioned responses. So there are layer 5 pyramidal cells that send their outputs not only to the facial nucleus and oculomotor nuclei and red nucleus, so very, very close to the output side, but also to the pontine nuclei. So you have this massive set of projections that descends from the motor cortex down to the pons, and I'll go into some more detail about that later. And it completes the loop. So there's a CR afferents copy that goes back up to VL thalamus and then back to motor cortex again. So moving on from that, that's the anatomy. What about the physiology? What are the Purkinje cells actually doing? And something that I find really very interesting is the idea that when you record from Purkinje cells in the eye-blink microzones during conditioning, and this work is in the decerebrate ferret. It's done in the work of Jerry Heslow, who's very well known for his work in this area. If you look at Purkinje cells before training, you can see that when the CS comes on, they just continue to fire. After training, you find a pause. So as soon as the CS comes on, these Purkinje cells shut up. They just don't do anything. There's an inhibition. After training, in fact, what they did here was that they switched from a short latency conditioned stimulus to a longer one to see what would happen. And they found that the timing characteristics of this pause changed to match the magnitude of the timing of that CS. You can see the pause a little more clearly in this figure here. One of the interesting things about it is that they reported that if you use gabazine, you don't find that this affects the response properties that I've just mentioned. Now, gabazine is something, it's something that inhibits GABAergic inputs into Purkinje cells. Purkinje cells are basically modulated by a very large range of different inhibitory inputs. And if you make them all go away, if you shut all of them up, you find that it is exactly the same. It doesn't change. And what does that tell us about the operation of the system? It says that these are independent. This decrease is independent of inhibitory inputs to Purkinje cells and can really only be affected by the excitatory input into these cells. So in other words, by the parallel fiber inputs primarily that are coming in. Okay, so it's the parallel fibers that are now causing this decrease. Why is this important to mention now? You're probably sitting there wondering at the moment, why is he still talking about classical conditioning when this talk is actually about instrumental learning and cognition and so on? I'm coming to that. But this gives us a handle when we're using fMRI. It gives us a handle on the kind of signal that we should expect to see when we do when we model the fMRI data. Okay. So let's now leave the narrow world of classical conditioning, the narrow but informative world of classical conditioning, and move into a slightly wider territory. I mentioned the motor cortex to you before, but of course cortico-cerebellar connections we now know extend well beyond the motor cortex. One of the first studies that shed light on this, and this is something that really shook the field up, was the study in 2003 by Kelly and Strick. Because for the first time, what they showed is that not only is there a motor loop, the loop that I mentioned before, that connects the primary motor cortex with certain lobules of the cerebellar cortex, but in addition to that, they used these very peculiar but useful viral tracers to demonstrate a transsynaptic connectivity that was isomorphic with this loop, that had the same structure as this loop, that connected the prefrontal cortex with a different set of lobules. So there are these two great loops. One we call the motor loop, the other one we call the prefrontal loop, the former connecting the primary motor cortex with one set of lobules, the latter connecting area 46. I'm not going to generalize very widely because area 46 is where they put their tracer. Okay, so they put their tracer into the mid portion of the principal sulcus in monkeys, and they found that there was label back here in CRUS 1 and CRUS 2. Here's a slightly more detailed figure. We know precisely which particular lobules receive and send these inputs. The other thing to note is that this information didn't only go one way. They used retrograde as well as anterograde tracers, and they found that whether you're looking at the efferent side or the afferent side of the loop, it was always the same lobules that were affected. And what this suggests is that the cerebellum, like the basal ganglia, is connected with the cerebral cortex in a series of parallel loops. Now, one problem that I have, and this is just a side story, one problem that I have is that it's never been demonstrated very convincingly that this really is a loop. It doesn't suggest that just because you end up in the same lobule that you also end up in the same neurons. So a problem that I still have with the literature is the point-to-point connectivity is missing. That evidence still has to emerge. But let's, for the moment, give them the benefit of the doubt. It's a plausible interpretation. Is it only area 46? Well, probably not. The work of Schmarman and Pandya has also been instrumental, and what they've found is that when they inject localized, small, very carefully placed label, I think it was HRP label, small injection sites into multiple prefrontal areas in monkeys, they find that there is a subset of these prefrontal areas that also project to the pons. Okay? Now, given that 99% of the stuff that ends up in the pontine nuclei will send its outputs on to the cerebellar cortex, it's reasonable to assume that these areas here in the frontal lobe of the macaque monkey project off to the cerebellum. And amongst them include areas back here, area 44 and 45, according to Petrides and Pandya, the same kinds of areas in the upper and lower banks of principal sulcus. And one of the most interesting areas for me is area 10 up here. And what they showed, and I'll come to later, is that there were also projections that cause quite heavy labeling in the pons.
And all these projections follow your standard cortothalamic organization.
Yeah.
They all run over VL, and they all originate then in the deep layers.
They all originate in layer 5. There's some discussion about which ones end up in different parts of the thalamus. They don't end up in motor thalamus. Some of them end up in MD, and so on, which makes sense because medial dorsal thalamus is, you know, the area that sends off to prefrontal.
But then you would also think about these zones that we see here.
Yeah.
these are the injection sites essentially. So in
That's
your
right.
mind, this is a continuous band of recurrent interaction with cerebellum, or you really see this as a zonal organization, very with discrete
You mean whether they're
boundaries?
discrete or well?
Yeah.
It's difficult. It's difficult. One of the issues, in fact, one of the most interesting things about Schmamann and Pandya's work is that they've shown that the ventral parts of prefrontal, down here, send very, very sparse or no projections. That all seems to come from dorsal prefrontal in the macaque. What that means functionally is an open story. I mean, there's the suggestion, for example, that, well, it's the dorsal areas in prefrontal that are slightly more concerned about action, even though it is higher level action, than these ventral areas that are much more sensory in nature in some ways. But I do see the dorsal aspect as being pretty much a continuous band, and I'm going to comment on that a little bit later.
Okay.
Particularly when you think about that band being itself hierarchically organized,
Right.
So let's look at the slightly more controversial side of this literature. I mentioned Mitch Glickstein, who I admire as an anatomist very greatly. He's contributed some fantastic work to the field. Where Mitch and I depart on areas in the literature that talk about anatomy is this. So in 2006, Mitch commented on the work that he published and the work that Brodal separately published in 1978 and 1985. So Brodal, back in the day, used degeneration as an anatomical method, and this is where you place a lesion in one part of the brain, the axons degenerate, and you use the silver method to pick up the target areas from which the degeneration originates, and you can then make inferences about the connectivity between those two areas. And what he did was to place lesions in the pons, and he found degeneration, not surprisingly, arising mainly from areas anterior to the central sulcus, that's marked out in red here on this macaque brain, and also some degeneration in the posterior areas here in S1, but not much else. Didn't find much else at all. And so Brodal's conclusion was that the cerebellum is processing information that's exclusively motor and a little bit sensory as well, but nothing else. Mitch did a different study. He injected the pons, he filled the pons, to use his words, with HRP, which is a retrograde tracer, and he then asked the question, if you section the entire cerebral cortex in these cases, where does the tracer end up? In other words, which are the areas in the cerebral cortex that send their outputs to the pons? And his conclusion was that the projections arise mainly from those areas in the planning of movement or the corollary discharge. I'm a cerebellar person, but I can still never pronounce the damn thing. From an intended movement.
That's why I always say efference copy. to
Okay,
sort of
yeah.
Make life easier.
If it was the same thing, I would, but I don't think it is. But anyway.
Okay.
So the other thing he said is that although there's a modest input from medial prefrontal cortex, there's very little or none from the lateral prefrontal cortex. Now, this has puzzled me for a number of years. And so I went back to his data and I realized that these are flawed conclusions. And this is why, you mentioned earlier that your students should be an extension of you, but I would say the opposite. I think my advice would be, if you're a postdoc or a student, you should question your mentors all the time. Make their life difficult as you possibly can. Having said that, I didn't do that, but it's important always to go back and not take anything as dogma.
Absolutely.
And the reason that I think these are flawed conclusions is this. If you look back at their paper, and you look at the sections, you can see the stippling is basically areas of cortex that send their projections to the pons. You can see that there's actually plenty of label there in prefrontal. Here's the arcuate, everything in front of it, lots of tissue there, okay, that contains label. If you take this particular section, just to illustrate the point in more depth, here's the principal sulcus, which is this sulcus here. You can see that there's very dense label in the dorsal bank of the principal sulcus, and lots of other areas too on the medial surfaces as well. So I disagree with Mitch, not just in principle in general, but on the basis of his own data, and he really hates that. We're quite friendly about it, but he doesn't like it.
But how does he explain that away?
He doesn't.
But he still disagrees with you.
He disagrees. This is. I think it's possible that people are set in their ways sometimes when they think about these things. But let's
Okay.
let's move on politely from that. Here's another graph from the paper. This is the number of labeled cells per millimeter. And what we find here as well, these are effectively the Brodmann areas, and we find that actually, if you look in medial prefrontal cortex, the amount of the cell density in places like areas 23, 24, 25 are the same as that which you find in the posterior parietal cortex. So I are not much lower than what you find in areas 4 and 6, which are primary and premotor cortex. So I simply don't buy the idea that the cerebellum is processing information that is purely motor in nature. There is an abundance of evidence now that completely defeats that interpretation. So in 2008, the story shifts slightly, and Mitch published some work with Doron where he said that the data are now unequivocal in demonstrating that there's a link between the cerebellar hemispheres, which project to the dentate and the prefrontal cortex. However, we suggest this connection may be part of an eye movement circuit. And this is, again, something that we should question and think about. And I tried to get to the bottom of this. I wrote to Peter Strick, who published the initial work in 2003, and I said, what's this? Did you inject in the eye fields? And he said, no, you must be bloody joking. No. I recorded before I injected, and these were not eye field areas. Anyway, you get a sense that there's some controversy around this, and some of the inputs from the cerebral cortex to the cerebellum appear to be cognitive, may be primarily involved in eye movement control. So again, I'll talk to you about an experiment in which I try to address that. Some of these prefrontal regions are cognitive in function, but some may be more closely related to the control of eye movements. So you can see that these conclusions are flawed in some ways, but it's important to have somebody like Mitch in the background poking and prodding us to make sure that we continually question our own way of thinking in these areas. So I mentioned Area 10. Some years ago, Adrian Owen and I got into a discussion about Area 10 and realized how little we knew about it. And we sat for 3 days continuously in the basement of Queen Square at the ICN, just poring over many, many, many piles of papers. And this eventually resulted in a review in Nature Reviews Neuroscience. And one of the conclusions that we drew in that review was that Area 10, according to some anatomists, in humans, is the largest prefrontal area that we're aware of. Of course, this all depends on how you characterize what an area is and so on. But if you take the cytoarchitectonic definitions of on-Goetherian price, you will find that this is a huge area in the human brain, especially when you compare it with that which you find in macaque monkeys. Okay, you can compare the two here and you can see that they're really quite different. And of course, this is an area nobody has recorded very extensively from there. The very initial recordings that were done by Aldo, who will be speaking to us tomorrow, along with Steve Wise. To the best of my knowledge, there are no eye fields in there. To the best of my knowledge, there are no outputs from this area that send their projections off to premotor areas or primary motor areas, or indeed send outputs to the spinal cord like some premotor areas do. But Schmarman and Pandya nevertheless injected some anterograde tracer into area 10 and found very dense labeling in the pons. And you can see that labeling appear here. So we know that the most anterior parts of the prefrontal that perhaps have the least to do with motor control, even eye movement motor control, are sending their outputs there. So that's all very well in the macaque brain. What about the organization in the human brain? One might still argue that actually maybe it's all different in humans. There have been 60 million years of primate evolution through which these things change. There's been about 5 or 6 million years of evolution when we departed from our most recent primate cousins. So perhaps there is some difference. And so we look at evidence from physiology here. We can't inject HRP into the brains of our undergraduates, unfortunately, so we have to make do with MRI. And what we did, along with Jill O'Reilly and Heidi Johansenberg in Oxford, was to define our cortical targets. So Jill defined a whole range of targets, including prefrontal and motor, and extracted the time series from these areas using resting state. And what they basically found was that the patterns that emerged in the cerebellum seemed to correspond very tightly with what was seen by Kelly and Strick in macaque monkeys. Okay, so what they found was that CRUS1 and CRUS2, these particular lobules here, contained activity that co-varied very tightly with the activity observed in the prefrontal cortex, and likewise for the motor lobules as well. So there's some correspondence between the human and the macaque, and we can start building on that. So I mentioned evolution earlier, and I think it's always important to study things in the context of brain evolution if you're going to study the brain. One of the most elegant and simplest ideas that I've read about is that of concerted evolution. If you think about the fact that selection pressures apply not just on individual brain areas, but they will apply on the entire functional system. Okay? They will apply not just to one brain area or another, but they will apply to an entire system. And there have been lots of papers out there that have argued that the prefrontal cortex has expanded very rapidly during the course of human evolution, particularly in relation to the motor system, which doesn't seem to have expanded quite as fast. So the question in my mind was, if the prefrontal cortex has undergone this massive expansion, can we expect that this whole prefrontal circuit would have similarly have evolved and expanded? Has the cortico-cerebellar prefrontal loop expanded more than the motor loop? If we want to frame it in a way that can be tested. So the work that we did to address that looked at two areas. We first of all looked at the outputs from the prefrontal cortex to the pons. And we also looked at the expansion of these prefrontal projecting lobules as well in various species. So I should say we looked in a number of different primate species. More recently, we looked at the cerebellar dentate nucleus as well, which I'll come on to. So here are these two loops again in a schematic way. Got prefrontal cortex going off to the pons and off to its own little zones in the cerebellar cortex, and likewise the motor loop. You can see that the cerebral peduncle is the fiber system, this great fiber system through which all these fibers converge before they arrive at the pontine nuclei. You can see also that the dentate nucleus is split into a ventral and a dorsal component, so it's possible to look at those separately as well. And that's basically what we did. Here's a figure taken from Gray's Anatomy. Now, another theme that I'll pick up on is the fact that you're sometimes supposed to throw your textbooks away. This is a figure that is still in the current version of Gray's Anatomy. And for some reason, being the obsessive person that I am, this just nagged me. And I thought, why have they divided up the human cerebral peduncle into these three zones? Basically what they were saying is the prefrontal inputs arrive through this small segment here. The bulk of stuff comes through this motor segment from the motor system, and then the temporal and the pontine fibers come from this tiny area here, back there. And I got to think, well, where's the evidence for this, for goodness sake? You know, what is the evidence for this? And I looked at the evidence, I dug it up. Of course, none of it's cited in Gray, so I had to go and find it in some other way. And it's all based on degeneration studies, which are inherently terribly unreliable. And there were some inherent problems with that work, and I couldn't let it rest because this simply didn't make sense to me in the context of the fact that the entire system should have expanded in concert. The proportion of motor fibers is huge, the proportion of prefrontal fibers is rather tiny, and that simply didn't make any sense. So the conventional view could well be wrong. So we used diffusion tensor imaging to try and understand the problem and get a better handle on it. And what this allows you to do, without going into too much detail, is to ask how fibers really distribute in the human brain. What is the trajectory of fibers that arise from the prefrontal cortex and from the motor system and converge through the white matter pathways of the cerebral peduncle? The other thing that it allows you to do is to segment the areas that are the targets. So this allowed us to segment out the cerebral peduncle according to where the fibers originated from. So we did this on 7 areas, not only of the human brain, but if it was going to be meaningful in the context of anatomy, we had to also do this in the macaque brain as well. So we segmented the human and the macaque brain into prefrontal, premotor, primary motor, and so on, into the various different zones. And we ran diffusion fiber tracking on these using Tim Behrens' method in Oxford, and what we found in the macaque monkey was very unsurprising. In the macaque monkey, we found this area shaded green here is tiny, and those fibers are coming down from prefrontal cortex. The premotor and the primary motor in the yellow and the blue were the next, and so on, and those were quite large representations too. So that's not very surprising. What surprised us is what happened when we looked in the human brain. In the human brain, we found this enormous representation in the prefrontal, coming from the prefrontal cortex. So this lends some support to the idea of concerted evolution operating in the cortico-cerebellar system, where a third of the fibers, assuming a uniform density, were coming from prefrontal cortex in the human, compared with only 15% in the macaque monkey. So here's some nice evidence that this prefrontal representation has expanded during the course of human evolution.
But was yellow, or I got lost now? So,
So the
the
yellow-
yellow bit is motor.
The yellow is premotor,
Premotor, okay.
and then you've got primary motor in dark blue and somatosensory in light blue and
Right,
so on.
okay.
But in 18 hemispheres, we found exactly the same pattern, this beautiful topographic organization in the cerebral peduncle. So my PhD student, Josh, was as stubborn as I was when I was a PhD student. And Josh said, well, look, you haven't actually looked at the cerebellar cortex. Why don't I go and do that for one of my thesis chapters? And my response is going to take you ages. You will never finish your PhD. And he said, no, I really want to do this. And we both took a slightly mad decision at the time that we'd let him do it. So what he did was to take 10 brains, MRI scans, I should say, from humans, chimps, and capuchin monkeys, so 30 in total. And he segmented the cerebellum into the different lobules in each brain, and he measured the volume. And what he found was that the motor lobules didn't vary very much in relation to each other. However, the prefrontal lobules is where we found the largest differences. So there was a much bigger difference between CRUS1, the volume of CRUS1 and CRUS2, between humans and chimps, and indeed between chimps and capuchin monkeys. The advantage of throwing capuchin monkeys into the mix is that they are New World monkeys, and so they provide a point of convergence that is quite far away from the very recent difference that has occurred between chimps and humans. And so again, we find some more evidence here for concerted evolution in the cortico-cerebellar system. The other point at which we can look is the dentate. The dentate nucleus is one of the three cerebellar nuclei through which the outputs of the cerebellum converge and exit the cerebellum. And I mentioned before that the dorsal part of the dentate connects with the motor cortex, but the ventral part connects with the prefrontal cortex, and the two don't meet. So Matano, many years ago, suggested that actually this portion of the dentate here in humans is vastly enlarged compared with not only macaque, but many other species as well. And so on the face of it, up till quite recently, that's been another piece of supporting evidence from the dentate. We didn't want to take that at face value because that approach has its criticisms. Along with Chris Steele and Arno Wilringer in Leipzig, we have conducted the first study that characterizes the outputs from each of these lobules to the dentate nucleus. And we use those outputs to segment the dentate. And what we find is that the motor lobules do indeed arrive dorsally in the human brain, in the dorsal parts of the dentate up here. And you can see that there's also a nice topography. But if you look here at the prefrontal lobules, their outputs are represented in the ventral parts of the dentate. And that's fine, that's as it should be. However, there is a very, very significant portion of the dentate is given over to the outputs from the prefrontal cortex. Or rather the outputs of the lobules to which prefrontal projects. Okay? So this is very preliminary work, this is still on the way. But again, it provides some support for the hypotheses about evolution that I've outlined. How are we doing for time, by the way?
Great.
Okay,
Don't worry.
fine. Let's leave anatomy for a while and reflect a little more about function. Let's talk a little more about what the cerebellum and the prefrontal can contribute to automatic and controlled processing in the brain. And I will explain a little bit in a few moments about the way in which all that anatomical stuff, how that impacts on our interpretation of theory. So those of you who've done some psychology will be aware of dual systems accounts of human performance. And the story really begins back in the 1970s with Schneider and Schifrin and many others who spoke of two modes of processing. So we have controlled processing that has a certain set of characteristics and automatic processing that has the opposite set. So controlled processing typically has limited capacity, needs attention, is very flexible, very serial in the nature of its operation, very good for online control, but very susceptible to distractors. And this is all beginning to sound, particularly from this morning's talk, this is all beginning to sound like a very kind of prefrontal type of operation. Automatic processing, on the other hand, few capacity limitations, typically doesn't need attention. The downside is that it's very inflexible. The upside, again, parallel in terms of its operation. Once you initiate these processes, they're feed-forward, so you can't really slam the brakes on very effectively, and they're much less susceptible to distractors. Okay, some of this is saying the same thing. Those of you who've read Thinking Fast and Slow by Danny Kahneman might recognize this. If you haven't read it, it's a damn good read, so go and buy the book. He calls the control processing system 1, and he calls the systems involved in automatic processing system 2. And what I'm tentatively suggesting is that system 2 could be run by areas like the prefrontal cortex. In other words, the areas that represent your goals, the higher level representations that need to be flexible. System 1, on the other hand, could run, could operate in the cerebellum. Now, I'm not excluding the possibility of the basal ganglia also being involved. A lot of basal ganglia aficionados will say, well, hang on, isn't that a basal ganglia characteristic? And I'm saying it doesn't necessarily have to be exclusively basal ganglia. So in any case, these ideas are partly spelt out in this Nature Reviews paper, but also in this more recent chapter that's appeared in a volume that I've edited in Progress in Brain Research. So a related idea, a related set of ideas is about control theory. So how does control theory work? Now, the reason I mention this here is that this is now very closely allied to cerebellar function. Lots of people have spoken about control theory in the context of cerebellar function. So I'm now attempting to stitch these two areas together. One of the other interesting and important things to do is to try and make sure that there is actual anatomical evidence for a control theoretic account of information processing in the cortico-cerebellar system. So typically you have a controller, a plant, and the controller sends a motor command to a plant, and the plant in this case is the spinal cord and the muscles. And that generates a movement, and when the movement happens, there are sensory consequences. And one of the things that the cerebellar cortex, it's been suggested, does is to read off a copy of the motor command and use it to generate some predictions. So it could predict, okay, what are going to be the likely sensory consequences if this movement is executed. It's then in a position to compare the actual and the expected sensory consequences of action. And if there's a match, then all is well with the system, everything's predictable. If there isn't, an error signal gets generated, and there's a correction that goes back to the primary motor cortex to say, well, you better update the way that you do things and the way that you issue your motor commands. I don't want to get into too much detail about whether this should be a forward model or not, and so on. I'm assuming that I'm assuming that this operates using forward models for the time being. Now, if you look at the anatomy of the system, it's very clear that the primary motor cortex doesn't operate on its own. It operates in the context of a chain of hierarchically linked areas. So we know that there is anatomical evidence that it receives input from the premotor cortex. We know that there's anatomical evidence that area 46, the area that I spoke to you about before, is sending its outputs to premotor. And we also know that there are lots of other prefrontal areas that send their outputs to area 46. So when you talk about a goal-directed action, it's not these areas operating alone. They are devolving, they're sending signals to lower areas in this hierarchically organized cortical motor system to generate the movement. But all that stuff that I told you about in terms of the anatomy also tells us this. Each of these modules has its own associated area of the cerebellar cortex that could potentially house a forward model. Okay, so we know that lobules 4 through to 6 receive inputs from the primary motor cortex. The areas of premotor cortex are not quite so well characterized, but they will have their independent inputs into a different part of the cerebellar cortex. We know about the connections from prefrontal cortex, area 46, that ends up in Crus 1 and Crus 2. And we've seen that there are also outputs from places like area 10 that end up in parts of the pons that will probably end up in different parts of the cerebellar cortex. Now the anatomical picture here is not complete, but that's the picture that seems to be building up. You can see also that the dorsal and the ventral parts of the dentate are differently involved. And if you dig a little further into the literature, you start to discover other slightly more interesting things, such as the fact that this system can be sensitive. The pathways exist for it to be sensitive, not only to the sensory consequences of action, but the decision consequences as well. Okay, so by decision consequences, I mean higher level things such as the receipt of a reward and that sort of thing, which is much more your territory than mine. But there are outputs from the VTA, there are outputs from dopamine-heavy areas that go back to the cerebellar cortex. Interestingly, there are outputs from the dentate that go back and they inhibit the VTA. They send inhibitory projections. And the structure of those seem to be similar to the structure of the outputs that go back to places like the inferior olive that deal more with motor control. So this is the system that I'm proposing that deals, this is what I'm calling system 2, and this is basically what I'm calling system 1 when it comes to dual process theory.
Sorry, one question.
Yes.
When you say that VTA connects to cerebral cortex, and on the other side you have inferior olive connecting to the cerebral cortex,
Yeah.
the properties of these two type of connections are going to be very different.
Oh, they're very different, absolutely. So, I mean, for a start, this is dopaminergic, and this is based on glutamate and one or two other exotic neurotransmitters, but it's excitatory in different ways. And one of the things that's really struck me is, what is the point of this inhibitory connection that comes back out from the cerebellum and tells and inhibits the olive? And this is a question that has been explored in the eye blink literature, and I've seen from a poster outside that I think you guys are probably exploring this issue as well. And if I can just speculate for a minute on that, I think one of the things that this system needs to do is, if you have a forward model running here, the last thing that you want it to do is to become suddenly responsive to error feedback while it's doing its job, right? So you don't want this system to slam the brakes on while there's and the whole point of a forward model is that the system just gets on with it and just executes a fast feed-forward output. So it's natural that then we inhibit the olive with this output. Sorry, yes?
Sorry. VTA, excuse me, if I will understand, VTA not only projects to the prefrontal cortex, but it does also to the vast premotor network.
Yes.
Yes, so the question is in your model here, if I well understand, VTA has its main input into that system. So you mean that it does through the cerebellar system one, or it does directly on the cerebral cortex?
Well, this is a simplified version, but yes, so of course there's no denying that the VTA will impact on all of these areas as well. But it's an alerting signal, and it basically tells this system, wake up, there's something funny going on. And you need a system like this to say, no, don't wake up actually, because if you wake up, you'll stop me. This goes back to the notion that you spoke of earlier about competing systems. And what I'm suggesting is that you have a system here that's competing with this system here, but nevertheless learning from it. So you need a system to silence all these other places that could wake up and get involved in online control under circumstances when you don't need online control to be happening at the time. And what I'm suggesting here is that the same thing happens with this output. So you have an output from the cerebellum that is silencing even the dopamine system and telling it to shut up. Okay?
So you see VTA as your switch. So you switch between system 1 and 2 via VTA.
I haven't thought of it quite like that.
But that seems to be
possibly
what you're saying. I mean, I don't agree with it, but that seems to be what you're saying.
Potentially, yes. Yes. And I have to also say that this is...
Don't worry.
No. The other thing is that these ideas are still speculative.
No, of
Okay,
course.
and there are bits of it that still need patching up. But effectively, this does provide a mechanism for the cerebellum to turn off the VTA. Yeah.
Okay.
And that's a hypothesis that I think
sure,
is...
it's cool.
Yeah.
I'm sorry to discuss this, but
No,
it's
sure.
becoming more interesting than what I expected.
Okay.
I'm learning the most today. But there is a known paper, hidden paper in the literature, not published in Mars or in the Moon. It's published in PNS,
Right.
where we did an experiment, Victor de la Fuente and I, a former student, and just because it kept in my mind from the beginning that Wolfram Schultz and I reported the first reward-related signals from dopamine cells. Hidden papers, 1990, Journal of Neurophysiology, twin papers, which has been lost in the literature. So I did the original experiment, and I was always struck by the fact that these neurons at that time were related to sensory cues that were always related to reward. So I did experiments in a decision task, and it happens that dopamine cells, which are in the PTA, they are related to perceptual decision making. They only encode the stimulus in a very parametric way, always that the monkey is right in its perception. And worse, they report also uncertainty and all what is required for decision making. So it probably, your model is probably right. There is a very tremendous vast network in between the cerebellar BTA and the prefrontal cortex, and of course, medial prefrontal cortex or the premotor network is related to decision making. I'm frankly very interested in reading the full anatomy paper on all your model, because certainly it's related, the BTA is related to perceptual decision making.
Right. So first of all, I'm very relieved to hear you say that. So you're suggesting that VTA is processing information about the correctness of the monkey's perceptions of the rewarding stimulus.
Yes.
Right. So that, I think, would fit with this interpretation. And I guess the thrust of what I'm saying is that these connections exist, and what we have to do is to stick some electrodes in there and just go and test it.
Now, the other question I have, you have decision consequence there impinging on inferior olive.
Yeah.
So how is that done anatomically?
Okay, so that's very interesting. There was a recent paper by Tom, I can never pronounce his full name, Roykrock, who's a well-known anatomist, and he has identified a set of projections that originate from the medial prefrontal cortex in the rat. And I admit these are rats rather than monkeys. And they end up in the principal olive. Mm-hmm. And the principal olive, as we know, is the area of the olive that sends its projections onto the prefrontal cortex. Mm-hmm. So when I say decision consequences, we know that the medial prefrontal, even in rats, has a role in processing the consequences, the outcomes of actions. And, you know, those neurons, it's not beyond the realm of possibility that those neurons impact on olivary function and convey that signal.
Mm-hmm. Okay.
I have a question about the two systems. What are the time scales of the two systems?
The time scales of the two systems, this is something that is not worked out.
Well, but wait, Kahneman
yeah.
is clear on this, right? That's why he calls it thinking fast and slow.
Yeah, so, okay, well, at that level of detail, this is the system that would be slow, and this system would be very rapid, and for two reasons. First of all, this is very serial, it's susceptible to feedback, and we know about all the feedback-related inputs that could end up in these areas. Feedback, for, I should say, for the purpose of online control. These systems, the physiology would suggest that they're very fast. The other thing is that, look, these are not connected to each other. You don't have to have these wired up to each other for them to work effectively. So these can operate independently of each other and very quickly. And so to that extent, if we're talking about precise timescales, I don't know, just so that I'm not being flippant. But crudely speaking, these cortical systems, these controllers and plants, would be operating at a much slower rate, I guess. Yeah.
Previously, you also showed that somatosensory cortex also had projections from the data in the plate. So where would it fit in
Yeah.
this diagram?
So where does somatosensory cortex fit into this? So this system that I'm talking about here is purely the motor system, and much of the sensory information that ends up in the cerebellum needs to get there very quick. There's not much point. I mean, it does receive inputs from places like the somatosensory cortex. We know about for example, the interaction between the whisker barrels in rats and the way that the cerebellar cortex processes those. But much of it is subcortical. The sensory information that arrives, so for example, the cerebellum, it's the head ganglion of the proprioceptive system, according to Sherrington. And the proprioceptive information that arrives into the brain, well, the cerebellum is one of the first ports of call. And for a system to operate fast and effectively, you simply don't have time, if it's for real-time control, for information to go all the way up via the thalamus, through to S1, S2 or whatever, and then send its information there. But there are projections that end up here for offline control from S1 and S2. Okay, so let's press on from that and talk about some fMRI studies that I've done. I hope we're doing still okay for time. This is a set of studies, a small set of studies that I'll talk about where I've used conditional motor learning. So basically this is where we go from classical conditioning to trial and error instrumental learning of goal-directed actions. And one of the ideas that I'm exploring with this is to try and understand how goal-directed actions that are run by System 2 evolve into habits that are run by System 1. So why are we using conditional motor learning? Well, I'm taking my inspiration here from Steve Wise, who has reported on this very extensively in primates. Dick Passingham has done a lot of work to follow this up in the magnet in humans. How does it work? Well, one of its big benefits, by the way, is that you can time lock activity to instruction cues and make inferences about rule-related rather than response-related activity in the brain. And it starts like this. You present the subject with an instrumental CS. This is just simply an instruction cue. This can be an arbitrary stimulus. In this case, it's just a red circle. There's nothing about that red circle that inherently tells you what action to perform. That's why it's arbitrary. For reasons that will become clear, we present a variable time delay, and the subject is then required to execute a motor response after being given a go trigger. And if they press the right button or they make the right kind of response, they get a reward. If not, they get a cross or something like that. So this is basically the structure of the trial. And one of the advantages of using an instrumental CS is that we could go back and say, and ask the question, does the cerebellum process information related to instrumental conditioned stimuli in a way that might be similar to the way that they process classical stimuli in the context of a classical conditioning paradigm? The key difference between classical and instrumental conditioning here is that the response and the outcome are contingent. There are contingencies between these three. In a classical conditioning paradigm, no matter how you respond, your response is always, the outcome is always going to be the same. Here you can modulate and change the outcome depending on the decision that you make. It's a very rule-related, very cognitive set of processes that are involved in the processing of these instruction cues. And that's also what makes it very interesting for trying to understand the role of the cerebellum in cognition. But when we do these things in humans using fMRI, the bolt signal is very slow and very sloppy and takes ages. And you need to tweak the experimental design in such a way that you can time lock activity separately to instruction cues compared with all the other events that are perhaps less interesting for this investigation. And the way that you do that is that you vary from trial to trial the interval between the onset of the instruction cue, which is on very briefly, and the onset of the response. And the result of doing that is that you reduce the correlation very significantly between the two. And when you come to model in fMRI, it's possible to make inferences about the activity that is time-locked to these instructions without any contaminating signal coming through from the other trial events, the go and the response and so on. So this blue hemodynamic response in these experiments is going to be related only to the instruction cue. There is no movement, there is no feedback, there's no go signal that's contributing to that. So a couple of years ago, Josh published a paper in the Journal of Neuroscience where I won't go into the deep details of the design, but the bottom line is that we presented our subjects with two kinds of instruction cue. And because of the way that we did the prior training, we were able to make them learn one much faster than the other. Okay? When I say learn, I mean they became automatic much faster than the other. So we pre-trained them at different rates before we scanned. And because of the way that we pre-trained them, they then became, these responses became more automatic. So more automatic, I'm becoming incoherent. Switch my prefrontal cortex back on. There was one set of cues that became more automatic more quickly than another set of cues. And we were able to differentiate between those, and we found that if we looked in the brain for signal that decreased, remember that we're looking for a decrease in signal here, signal that decreased faster for the more quickly learned cues than for the more slowly learned cues, we found an area of the cerebellum in CRUS1 and CRUS2. The other thing to bear in mind that we used only the correct trials. So another advantage of using event-related fMRI is that you can throw out all the trials where they were making mistakes and errors. So this is a very pure signal coming from correct trials where there's the area here is responding differentially to faster and slower learning. And so you can see that the trials where they were learning more quickly decreased in amplitude more rapidly than the trials that were being learned more slowly. So that's a nice demonstration that the conditioned stimuli were showing a pattern that is perhaps similar to the kinds of things that people have been observing in classical conditioning. And so maybe there's some scope to compare these two. Yeah.
Cerebellar learning has like very short time constants, right?
So it's so to learn like associations up to 500 milliseconds, 1 second maximum. So in this paradigm, you have like a varying delay.
Well, no, there's a yes. I mean, you're right in the sense. And when you say it has a short time constant, what do you mean?
I mean in classical conditioning, the association between the conditioning stimulus and the unconditioned stimulus.
Yeah.
would hold
There's a
up
brief
to-
gap.
Yeah, exactly.
Yeah,
And would hold like in a window up to 1 second.
That's right.
When you
Yeah.
tear those two apart, that you would not learn.
I think that's a really interesting point. So in classical conditioning, you have an interstimulus interval sometimes of
Yeah,
even up to 500 milliseconds.
exactly.
But this is obviously a lot longer. The answer to that is I don't know. I will say that we've done some recent work looking at working memory delays. And we find delay-related activity that lasts for several seconds in human cerebellar cortex. So my question would be, has anybody looked at these sorts of delays? So for example, you know, the work of Goldman Rakish from decades ago, where they found the memory fields with these long delays. Has anybody ever looked in the cerebellum to see if they're there? And my answer to that is probably not. So they may well be there, but I think it's an empirical question.
Yeah, because at this point, like, either the cerebellum has different time constants according to the different, let's say, areas of the cerebellar cortex, or some external structure might intervene in that.
Yes.
So
yes.
sometimes, like, the hippocampus is like involved in like classical conditioning type of setups, no? So where
Yeah, so the trace
it holds
versus
like the
delay
trace
literature and
somehow,
all that.
etc.
Yeah.
So
Yeah.
what hypothesis are you
Oh,
more?
you know what? I don't know.
Okay.
And I think it's okay to say that. But I think it's possible that prefrontal cortex is working in concert with cerebellum in this instance and not working purely independently of it.
Okay.
And maybe that's, maybe there are prefrontal areas that are holding this information over the delay period.
Thank you.
Sure.
But did you actually look at hippocampus or prefrontal in this task?
We didn't find any hippocampus coming out, and we did find prefrontal. We found, I think, similar decreases in prefrontal cortex as well.
Yeah, but it depends with how you do the contrast, right? Because for hippocampus, it might not matter that you learned about this stimulus or not.
Mm.
It just wants to hold it then
Yeah,
in a buffer.
yeah.
So then you wouldn't see it, right? Because it doesn't modulate
Oh,
it.
that's true. That's true.
So
You would
it
see
might be nice
something
to have-
related to the you'd see something related to the onset. I mean, we're not modelling in this experiment. We're not modelling what happened during the delay period. These are transient responses, and you'd expect the hippocampus to at least produce a transient response,
Mhm.
and we would have seen that, I think.
Okay.
But with F-
Because you answered Giovanni by saying maybe people should look for these longer
Yeah,
duration
yeah.
responses. Alternatively, you might already have the data in your hands, at least
yeah,
tick off the hippocampus
That's
and
right, but
prefrontal from that list.
I think the critical test, although I use fMRI, if I had a chance to do this with electrodes, I would do it with electrodes. I think the initial evidence like that, I think it would be better coming from electrophysiology, and the advantage is that you then have a targeted area.
Right.
You
Well,
look for-
Ricardo was in this experiment with Gary Heslow, and he's been holding Purkinje cells for hours and hours on end. So if you've ever seen a Purkinje
okay.
cell holding a response for longer than a second,
We never tried also, but
no, but still.
from the lab that I know, no.
Mhm.
They tried, but it was not holding more than 500, 800. 800 looks like the limit, but yeah.
Milliseconds, right?
Where were you recording from? Oh, probably from the Heimlich zones, right?
Yeah, from my areas, so this is 3 and
Yeah.
In ferret, right?
infrared.
Yeah. But you see, in the decerebrate ferret, in the decerebrate ferret.
Yeah.
Yeah. So I mean, one of the issues is that maybe you're recording from the wrong kind of place. You need to record from a cerebellar area that is actually wired up to areas like the prefrontal that can hold this information over long periods. So maybe moving. You'd have no reason to do it, but if you did move your electrodes into the lower lobules, into posterior lobe, and look for sustained activity in areas like Cruce 1 and Cruce 2 in this sort of task, then, and it could be that you're looking at the wrong species as well. I mean, doing this sort of thing in ferrets, I imagine, would be difficult, particularly if they're decerebrate ferrets.
Right. Okay, it's clear.
but
Narender
yeah.
doesn't want to check his hippocampal data. So
Fine, okay.
Fine.
Right, so that's that. The other thing, for the sake of time, I'll skip over that and move straight to this one.
Okay. Don't worry.
Yeah? Okay, well, let's talk about this a bit. So the other thing that Josh did was to do a similar study where he looked not only at those first order rules, the ones that I was talking about earlier, but he also looked at higher order rules. So he decided to look at rules about rules and compare them with rules about actions. And the reason that he did this is that it takes you even further away from action-related representations, much even further into the domain of cognition. He found, again, very similar effects. He found these second-order rules seem to activate similar areas of the cerebellum. They weren't the same areas but they were similar areas of CRUS1 and CRUS2. More recently, Georgios Argyropoulos, who was a postdoc in my lab, along with Jennifer Mills, who's a PhD student, undertook a fairly ambitious project where we tried to do the same kind of experiment using eye tracking in the magnet. And I don't know if you've ever done eye tracking in the magnet, but it's fiendishly difficult. And I have to give both of these guys enormous credit for getting it up and running. So again, this is how it worked. This was, again, an event-related fMRI study where we attempted to time-lock activity to an arbitrary conditioned stimulus. There was a fixation cross, a 400 millisecond instruction queue, a variable delay, a go signal, which was the disappearance of the fixation, a response, which is a saccade to a target that was arbitrarily selected. We then had another variable delay followed by feedback, and so the target would go green to say you got it right, or it would go red to say you got it wrong. There were a number of different variable delays here which allowed us to time lock to all kinds of activity, all kinds of stimuli, all kinds of events. But what I'll report here is what happened when we time locked to the instruction cues and the response signal. So after an intertrial interval, the next trial would repeat. There were basically three kinds of trial. We had a control trial that was simply an arrow instead of an arbitrary cue. And the value of this was that they would execute, they'd be presented with a very similar visual stimulus, they'd execute exactly the same response as they would on other trials. All the elements of motor preparation and that kind of thing would be controlled for. The other types are that there were two trial types. One was a set of fully reinforced instruction cues where they were always given viable feedback, and another partially reinforced. I forget what the reinforcement schedule was, but it was partial. I think it may have been 40 or 50%. But you can see that there was a clear learning curve that depended on the kinds of feedback that we were giving them. So the first thing to say is that we wanted some internal validation of this study. We wanted to ask the question, when you actually make a movement, forget about all this other stuff, when you actually make a movement, do you activate the oculomotor system? And the answer to that was yes. We were very relieved to see activity here. This red blob that you see here is the oculomotor vermis. We also found activity in the supplementary eye fields and the frontal eye fields, and I think also a little bit in the superior colliculus. We then asked what...
Wait, question.
Yeah.
The activity you see here, is it modulates up or down? In
This,
terms of the neural activity, what do you expect? Because what
well,
you measure the blood
yeah.
flow, right? So
that's right. So this activity goes up. Now,
Okay.
I don't know. It just goes up.
right. But
And with fMRI, you know, you could. Would you predict it would go the other way, or what would you say?
Well, actually, you showed us earlier fMRI on humans where you showed that a significant part of your data was that it was diminishing.
That's
and it
right.
would reflect that these Purkinje cells are pausing because they do
Yes.
something meaningful.
Yes.
Right, this is the trick of Purkinje cells. So then, if these guys would be doing something meaningful, you would say, well, then if these guys are involved in trigger response, we should see them diminish in their response, which would correlate with a diminished blood flow measure.
Right. So you're suggesting that you'd expect to see an inhibition here.
Yeah. Wouldn't you?
You would. So what the logic is that you'd see inhibition here that would lift inhibition from deep cerebellar nuclear neurons and
through that
cause
trigger an event.
exactly. Now the issue of what causes that increase. Don't forget, this is not neural. This is metabolic.
Yeah. Sure.
And we could be seeing the additional influence, the additional involvement of interneurons. So if you have interneurons kicking off here causing neural inhibition, then what you'll expect to see from fMRI is an increase.
Sure, but you earlier showed the Heslow paper
Yeah,
where they actually blocked the GABA.
yeah,
And this has no effect on the learning,
right.
right? So that explanation doesn't hold.
But this isn't a learning-related signal. This is simply people moving their eyes to a target.
Okay,
Yeah,
but you see
I
the
mean-
problem, right?
I see, yeah. I mean, typically what happens when we look even at people moving their fingers in the magnet, you press buttons in an fMRI scanner, you see these massive increases in activity. It depends a lot on whether it's an event-related design, whether it's a block design, whether, you know, there are all kinds of things that could influence whether it's increase or decrease. But I take on board your point that under a learning-related scenario, you would expect to see a decrease in signal here.
Mm-hmm.
But these are simply time-locked to the point at which they make an eye movement. And whether you would continue to see a decrease in the cerebellum under those conditions, I don't know. And I don't know, perhaps people who've recorded in the cerebellum could comment on that. But in answer to your question about learning, we looked at instruction cues and we did indeed see a decrease in signal for the FR and PR cues compared with the control cues. So it appears that for the learning-related activity, we do see these decreases. And we don't find it for the control, and we don't find it for the movement. So just to reiterate, when we time lock to the instruction cue, we find that activity in lobule H7A in cruise 1 and cruise 2, which are areas that project to prefrontal, decrease in terms of their activity. Okay, so that again seems to tie in nicely with this idea that no matter what kind of CS you're talking about, whether it's in the context of instrumental, whether it's in the context of classical conditioning, it seems to evoke a decrease in activity in the cerebellum. So over a range of different studies, we found activity that's in a remarkably similar area of the cerebellum. I've reported 3 of these studies to you today. There are another couple of studies that show broadly the same thing. So the thing that they all have in common is the activity that's time-locked to a cue that's been overlearned, that's been overtrained in an instrumental context. So in conclusion, we've shown that there's an expansion of the prefrontal cerebellar loop. So the pathways that connect to prefrontal with the cerebellum, they do exist, and that's been a controversial point in the past, but not anymore, I think. They do exist, but they've expanded during the course of human evolution, and they have greater significance in the human brain. In terms of physiology, I'm suggesting that there's an event-related decrease in CS-locked cerebellar cortical activity, irrespective of whether it's classical or whether it's instrumental. And finally, in terms of behavior, I think we have a system here for supporting the acquisition of skills and habits. And with that, I will, before I close, I'll just shamelessly plug a book on cerebellar learning that's been produced. I've edited this, and it's been a privilege to work with these co-authors here that include Ito, Egidio Angelo, a whole range of people who work on cerebellar learning. And finally, also, there's a short-term postdoc position in my lab. If you know people who are interested on working on cerebellar learning, please come and see me. The application deadline is around the corner, so get your skates on if you're interested. And finally, and most importantly, I want to thank the people in my lab who I wouldn't have been able to have half the ideas and do even half the work without their input. So I want to thank them, my collaborators, and my funders. So yeah, thank you very much.
All right, fantastic talk. Questions? Ivan.
So thanks for the talk, very interesting for me. A very basic question. So a problem I have sometimes discussing about cerebellar learning with people from different backgrounds or experimental, I don't know, is that you have talked now about habits and how cerebellum automatizes learning, and this normally is a slower process. No? So you have first explicit acquisition and then you automatize that. But then people that in the movement adaptation literature, their cerebellum is supposed to be the fast system that rapidly adapts to these perturbations in movement. So I never know what to say about that after that.
Well, I think the simple answer is, what is the evidence that it's a fast system? What is the evidence that it adapts very quickly?
Well, I think, I don't know.
Well, I don't think there is.
It could be possible that both things are correct. No, that it's slow for this habits acquisition and fast for movement adaptation. I think there's evidence for cerebellar patients.
Well, I don't know. I mean, is it fast for movement adaptation? I don't know that it is necessarily. I mean, if you think about classical eye blink conditioning, it's very slow. You know, if you think about the acquisition of any kind of reflex, it's typically quite slow. Even anecdotal examples, learning to ride a bike, at least in my case, very slow. All of these things, they are slow. It takes time from trial to acquire these things.
For instance, I think in the book and the volume that you have shown, there is work from Michael Jordan, from adaptation, in adaptation to force fields in reaching. I think there people with cerebellar damage don't adapt, and adaptation is fast. So there is a mix between.
Well, you've got to think about the fact that they're using two different systems, and one of these systems is damaged. In the case of patients with cerebellar lesions, you have to remember that those two systems, systems 1 and systems 2, they can probably operate in parallel, and they can probably operate independently from each other. There's redundancy there. If you have a lesion in the cerebellum, then maybe you're forcing system 1 to get on with it. And the prefrontal cortex is a pretty flexible system. Do you buy that?
Yeah, I think that's the kind of answer I was.
So there are other personal problems you want to share with our speaker? Okay. So, so the point is, but we shouldn't confuse ourselves here about fast and slow, right? Fast means that in terms of the real-time operation of the system, this is fast. It operates in a very short time with a short time constant for immediate automatic control. This doesn't imply it must be a fast learner as well. So we should be very clear about what's the time constant of the acquisition of this automaticity, and what is the operational time constant.
So exactly.
You have fast acquisition, and for automaticity you have slow acquisition attributed to the cerebellum, or that the cerebellum is necessary in these acquisitions, and in one case is fast and the other is slow.
Are you happy with that?
Okay, I'll buy it if you do.
He's hungry, he wants to get out of here,
No,
but
no.
not so fast.
So again, thank you for the beautiful talk. Peter Strick has shown also that the PFC is not the only area that projects to the lobules 7 and crus 1 and crus 2, but there's also the basal ganglia in the loop.
Absolutely.
Yeah. So my question is, like, do you see like all these circuits like somehow like interconnected, or they will be like two parallel systems?
You raise a really, really interesting point, and something that has not been addressed adequately in the literature. And the point is simply that in the past, people have discussed basal ganglia and cerebellar systems. I mean, they first discussed these systems when they talked about the basal ganglia as being a series of strictly operating parallel loops. And then Peter Strick applied the same idea to the cerebellum. As I said before, first of all, there isn't a huge amount of evidence showing point-to-point connectivity, and that these really are absolutely, strictly speaking, loops that connect individual neurons. So I think there's been a tendency in the literature to overgeneralize and to try and oversimplify things. What you're alluding to is that you may have basal ganglia and cerebellar systems interacting.
Yeah, exactly.
And you simply can't get away from that possibility. And as I've said in, let me just go back a few slides here. Sorry about this. Back to this really awful slide. There are basal ganglia systems that do project to the cerebellum, and they do share information with the cerebellum. The big question is, what is different about these systems? Because they're very similar anatomically. And I think that's an open question. The anatomy is very similar. In some ways, the physiology is very similar. You know, if you compare the medium spiny neurons in the striatum, they seem to be wired up very similarly to Purkinje cells. And it's been a big debate in the literature for a long time about what is different about these systems now. And yeah, open question.
Also like what one learns from the other, because like you have like projections from STN through pons in the cerebellum that reverses more or less the same area of PFC.
Yeah.
And then projections back to the striatum. So this is like somehow puzzling.
Yeah, they're systems that learn from each other, perhaps. Yeah.
Okay, but do you think this will be somehow involved into reward prediction errors or something like that? So some sort of like circuit that would involve
I think
also
where it could, I think the way to start solving the problem is somehow to go back to the motor system, because it's a system that we understand the most. And one thing that the basal ganglia seems to be well understood for, and in contrast to the cerebellum, is its ability to select actions and to sequence actions. The literature about cerebellar engagement in that is much, much less clear. And maybe that's a starting point where we can start to study the differences.
Hi, sorry, was there a way to differentiate between the white matter and the grey matter maps? Because you did both fMRI and DTI. So I was wondering, or at least, I don't know, in the macaques and the humans and stuff.
So basically going back to the DTI, are you talking about this study or the previous one?
And all of them. Oh,
Okay,
because I'm
so
assuming.
DTI in general.
Sorry.
That's all right.
I didn't quite understand here what kind of maps we were looking at. So was
okay,
it?
So this one here. Let's just go back here. We used diffusion tensor imaging to do this, and we basically. Are you doing DTI? Are you?
I've learned about it. Sorry, I thought DTI looked at white matter. Maybe.
Yeah, it did. Absolutely. Yeah. So the point is that we look at the trajectory of the white matter fibers. There are various things you could do to, for example, the FA maps and so on, to work out the trajectory of those fiber pathways through the white matter. The grey matter doesn't really come into it.
Yeah, so my question was basically because, well, it's quite basic, but white matter fibers are faster because they transmit the signal faster. So maybe if you, well, this is too far-fetched perhaps, but if you construct maps with that are composed of individual fibers that are faster, you get like a faster map. You can't have such a thing as a faster map, but like if you put it in time, say, as an animation kind of, I don't know.
I think what you're trying to say is, have we differentiated between the trajectories within grey matter and the trajectories within white matter?
Yes.
The answer to that is no. But I think the fiber, I mean, people make very grand claims about DTI, and they sometimes make the claim that it's possible to look at anything. My personal view is that the fiber geometry of things in white in the grey matter is probably too complex still that we can reliably study it. I think this sort of technique is very good for big bundles of white matter like the cerebral peduncle.
So you've told a very nice story about the parallel coevolution of the prefrontal cortex and the cerebellar cortex. But then you've told us this other story about these being two distinct learning systems, and I feel there's a little bit of a contradiction there. You know, the fact that they've evolved together suggests there's, you know, the prefrontal cortex and cerebellum are operating very much together to solve certain kinds of problems.
I don't think that it would be a contradiction. I think let's just, if we go back again to this hideous diagram. Yeah. Okay. Well, fine, but I think it could be. I have to be self-critical. I could be fair enough. You're suggesting that this system, because this system is operating in a very different mode from this system, that somehow contradicts the coevolution story.
Well, the connection between the two doesn't seem to be as essential if they are operating in different ways. So you could imagine maybe some animal which very much has developed the fast system and less the slow one, having a big cerebellum, for instance.
Well, I think in some ways these systems are also, I mean, they're competing, but they're also interdependent in some ways.
Yeah.
The reason for that being, let's say you have a system 2 without a system 1. You're going to be entirely reliant on the disadvantages of controlled processing over and above automatic processing. If you imagine getting rid of system 1, you're stuck here with something that is very flexible, but it is very slow, and it is you have to keep applying attention to do all of your problem solving. The advantage of this is that you can farm off some of the tasks into system 1, and they can run in parallel with each other. So this system here effectively frees up capacity in this system here. And so to that extent, they may well be interdependent.
Do you know of some sort of studies where people have intervened in the cerebellum? I don't know, maybe using
TMS perhaps.
Yeah, TMS would be a nice example.
Yeah, and so what tends to happen, I think they've tended to look at motor control more than this sort of thing, but we've done a bit of preliminary work where we show that there is an effect. So if you basically apply direct current stimulation over the cerebellar cortex, you slow the information processing up here, so your reaction times slow down, you start to perform more errors and that sort of thing. And that could be that this system is sort of overloaded.
Or it could be that maybe the cerebellum is providing, because the other story that is often told is that the cerebellum is the millisecond timing. It's part of the brain, and the basal ganglia is second timing, and prefrontal is maybe more about planning.
That absolutely. So that timing literature is, I think, it's not very coherent in some ways. But the other problem with studies of interventions is that if you mess up any part of one system, you'll inevitably mess up the physiology in the areas that it's connected to. So interpreting a transcranial direct current stimulation study or a TMS study, if you impair the fundamental processing in the cerebellar cortex, you're bound to find changes in connected areas, the physiology of the connected areas in places like prefrontal. So it's difficult to draw any kind of conclusion from those sorts of intervention studies, I think.
Hi, just to expand a bit on this relation with the basal ganglia and cerebellum. I think someone put forth a notion that the basal ganglia is like a reinforcement learner and the cerebellum is a supervised learner.
Yeah.
How do you think it fits into this? Or what are your views of this notion?
I think that's a very, that's a conventional view. But if you look at the wiring of the cerebellum and if you look at the physiology of the cerebellum, there's no reason why it can't operate as a reinforcement learner from that point of view. So these are quite traditional ideas. But the wiring diagram suggests otherwise, and the physiology suggests otherwise. So my view would be that, if you had to test both systems in the same way, that's what you've got to do to try and demonstrate that. I don't think those experiments have been done. I don't think there are enough experiments that have looked at the role of the cerebellum in instrumental learning, for example. So we can't rule that out, I think, still.
So about your beautiful diagram, how does System 2 train System 1 exactly, and how then is System 1 able to execute these acquired habits?
That's a really good question. If you look at the motor system as an example, so let's go even simpler and we look at come on, there you go. So if you look at this system, a really simple system for the eye blink, how does System 2 train System 1? Let's look. I mean, basically, the same.
We don't need System 1, right?
No.
Because we're just fast.
No, but in the case of some instrumental forms of this conditioning, I would sort of predict that motor cortex would get involved in that. But if you go back again to, this is getting silly.
No, you're getting pretty fast.
That's my cerebellum in good nick then. So you're asking how is this system trained by this system?
Right, because System 2 is deliberate. It's trying to figure out the problem. It says, okay, I go left and right and through the door to the fridge, get my beer.
Yeah.
Right? So great, but it's slow. So it takes forever to get your beer. So now it has to train System 1 to make it a habit, so you can sort of habitually grab your beer as you think about your next experiment.
Sure. So it's the development of forward models in these units that relies on things like efferentoscopy. Yeah.
So all my System 1 knows about what System 2 is up to is the efferentoscopies that I receive.
The efferentoscopies, but also the feedback that's coming back up. And I think your question earlier was it was related to feedback, is that right? So how does the system know that it's, no, your question was how does it do something about it?
Decision consequence, how do you get it in there?
Right, the decision consequence, we know about the fact that the dopamine system is very heavily involved in processing decisions. So lots of the work that Ranulfo's done, Wolfram Schultz, for example, has shown that the outcomes of decisions are coded in terms of an error signal from dopamine and so on. we know also that there are systems that project into the cerebellar cortex from medial prefrontal cortex, and we know about medial prefrontal responses to those sorts of errors, decision-related errors. So we know that the role of anterior cingulate, for example, in processing those sorts of outcomes. So we know that the wiring is there to process those inputs. So your question about what does the cerebellum, how can the cerebellum do anything about it? So how do these areas end up controlling behavior? Again, an open question, but the pathways exist for them to go back and modulate these parts of prefrontal in much the same way that these outputs from here go back and influence activity in the motor cortex.
Okay, but because the thing is, system 2 must train system 1, right? So, and then what you're saying is, okay, the wiring is there to do it, dependent on something like a dopaminergic reward signal, but then when it plays out the habit, it will play it out over primary motor cortex.
Yeah.
That's your strong statement on that.
That's
it
what
wouldn't
I'm...
go out over my pedunculopontine, motor nuclei, or red nucleus, or anything like that.
I think in the motor system, it probably will, because the wiring exists for that. But for prefrontal, we don't have that. There are no connections between these parts of the cerebellar cortex and that go out to the motor system in quite the same way. So their ability to influence behavior is really limited to their influence on places like this.
Very good.
Thank you.
If you have no more questions, then thank you very much.
I'm going to introduce our last speaker of the week, which is Greg Riccanzone. I've known Greg for more than 20 years. He was a graduate student in Mike Merznick's lab when I was a graduate student in John Kaza's laboratory. And we've been, since 1995, together at the Center for Neuroscience and have been collaborating for almost 20 years. So most of this week has been about development, comparative work, evolution. Greg's going to talk about how the adult brain changes, which for many, many years after the discovery of critical periods was like, okay, here are the critical periods. This is the time your brain is highly plastic, and then you're done. You're hardwired. But he did some incredible groundbreaking work with Mike Merznick in the 1990s, looking at how the brain changes with skilled use, and has continued to do incredible work on how the brain changes throughout the adult lifetime. So his title of his talk is Cortical Plasticity Throughout Adulthood, Implications on the Evolution of the Nervous System at the Nanosecond Scale. He did say he's kind of bummed he doesn't have any cool pictures of octopus in his talk, but it could be worse because he could be giving a talk behind a Nobel Prize winner who's had cool pictures of octopus in his talk right before cocktail hour. So it's not hopeless.
Like Leah said, we've known each other for a long time, and I was a little bit worried about your introduction because you have so much dirt on me that it would be, but I have a whole different set of slides in case you did do that, because I have a lot on you too. So we do a Mexican standoff. Okay, so thank you, Leah, for the kind introduction and invitation, and I'd like to thank Paul and Tony, and especially Anna, for all you guys have done to put this meeting together, which has really been terrific. Last year, Leah asked me if I wanted to do this, and I said, I don't know anything about development or evolution. I can't do that. And so I didn't come. And then she came back, and for like 2 months, that's all she talked about. And then she invited me again this year, and I thought I can either come and have a great time and do this, or listen to her talk about it for 2 months afterwards. And so the choice was pretty obvious. So here I am. And she asked me, she said, I said, well, what do you want me to talk about? She said, well, students these days, they don't get too much information about cortical plasticity. Cortical plasticity is a given. Everybody knows that it happens all the time. And so maybe it would be nice if I gave a little bit of the history behind it, and then talked about what she was saying, some studies that I did with Mike Merznick on how you can change the brain and how that influences the percepts. And then I'll talk about a question that really bugged me when I first started doing this, and that's how do you represent perceptions in a non-topographic piece of cortex? And most of the interesting cortex, if you think about it, and which is thinking, is non-topographically organized. And then I'll kind of finish up and talking about what happens in a normal brain as you get older, okay, so much older, and finally try to tie this all together and see if I can't make some inroads into what this means for development and evolution. Okay, so we'll start with a brief history of cortical plasticity. So it's not brief in the sense of time, really. I'm going to start with William James, who wrote the Principles of Psychology in 1890. And in that, he was describing learning. And he said in there that adults are able to learn new skills and behaviors really easily. And it's ubiquitous. And in fact, it's so common that no one feels the need to actually try to understand it or to explain it, right? So back in the 1890s, our brains were very plastic, which is given by the fact that we can learn all kinds of new things. And then a little bit later, Sir Charles Sherrington and Thomas Graham Brown did a series of experiments in the motor cortex of monkeys and apes. And they have this really cool paper here on the instability of the cortical point, which is published in 1912. And what they show in that is that if you microstimulate a brain in the motor cortex, and you do that for a while, you actually change the motor map. Okay, so you can do this over the course of minutes. The reason I learned about this is some 75 years later, Randy Nudo did exactly that same experiment and thought he invented this or discovered it, and then did a good literature search and found out that he had been scooped by 75 years. So the moral of that story is if you find something cool, don't go look it up, because you'll find out somebody did it a long time ago. Then there was Carl Lashley, who spent a lot of time trying to find the engram. He was looking for, when an animal learns something new, what part of the brain changes that gives rise to this new behavior, this new knowledge, or this new learning. And he's got this great quote, which he said near the end of his career, which is, "This series of experiments has yielded a good bit of information about what and where the memory trace is not. I sometimes feel, in reviewing the evidence on the localization of the memory trace, that the necessary conclusion is that learning is just not possible." Okay, so he just couldn't find it. All right. So all this was going on until Hubel and Wiesel, we've seen this picture before, and they did the landmark studies that showed not only how cortical columns were organized, but also that there's a critical period. As Leah said, if you, because of these guys, it really revolutionized the way neuroscientists thought about how the brain worked. Okay. All these neuroscientists' brains were changed by reading these papers, right? And they realized that their brains can no longer change, right? That was kind of the odd thing about all that, right? Okay, so yeah. So anyway, there were a couple of clowns around that time that got together, and that would be Mike Mersnick and John Koss. So Mike was a postdoc for Jersey Rose at the University of Wisconsin, while John was an assistant professor there. And they did a bunch of experiments together and ended up forging a lifelong friendship. And then Mike, being the way Mike is, decided he'd go to Vanderbilt on sabbatical, and they did a bunch of studies on owl monkeys. Okay, and this is what an owl monkey looks like. We've heard a little bit about them from Barbara before. They're a nocturnal monkey, and there was a couple of reasons that they decided to look at the somatosensory cortex of the owl monkey. And I should note that after Brian's talk, these monkeys, in those days anyway, they were all wild caught. Okay, so they have all the normal kinds of upbringing, rearing things that happen in owl monkeys as opposed to, you know, laboratory bred or whatever. I always thought that that was a bad idea because basically these are the monkeys that were dumb enough to get caught. So all these studies are on dumb monkeys, okay? So that's maybe not such a good reason to use owl monkey. But a really good reason to use them is their hands. Okay, they have really elaborate, nice hands, and they're nice and small. And the main reason really is because of their brains. Okay, so here's the lateral view of the owl monkey brain and dorsal and ventral view, and they have a nice lateral sulcus, but really no central sulcus. So if you compare that to a macaque monkey that has a really deep central sulcus, where the primary somatosensory cortex is down inside of that, right? And the owl monkey, okay, so it's down in here somewhere, yeah, down in here somewhere. And the owl monkey, this is central dimple, it's called, and the hand representation is right here, which is what Mike would call out in the flat. Okay, so you can take your electrode and you can anesthetize the animal, expose the skull, remove the dura, take a picture of it, and then you can see where you stick your electrodes and do what's called a map. Okay, so how that works in practice is you open up the skull, you take your electrode and you stick it into this spot right here, and then you record and ask, where do I have to touch the skin in order to get a response, right? And the verbiage there was just visible skin indentation with an opaque glass probe. Okay, what that means is you got this little piece of blue glass, and you just touch it as lightly as you can feel it. Okay, so it's really light. And so if you do it here at 0.1, you see that's the very tip of the pinky finger is what all you have to do. That's the receptive field. Then you pull the electrode out, stick it into point 2, and you see it's still on the pinky, but a little bit more proximal, and say 3, 4, and 5, and it marches proximally down the digit. If you go across from 1 to Q to A, you see that the receptive fields march across the tips of the digits. And then you get this map. Okay, so it takes, all night to do this, but at the end of the day, you can draw lines around the digit representations and see what it all looked like. Okay, so there's another important point about this, is what you notice is, so see G and H over here, which are these two over here. H is completely on the third digit, and G is completely on the second digit. It's really rare to find receptive fields in this kind of map of area 3B that were on both digits. Okay, they're almost always on one or the other. So Mike and John do this for a bunch of monkeys, and they get maps that look like this. Okay, so at the top here is the 3A, 3B border. 3A is cutaneous representations. 3B, or 3A, is non-cutaneous. It doesn't respond to just the visible skin indentation. You have to mash on the muscles or something like that, or move the joints, right? And this is the cutaneous part. And there's two ways you can look at this. One way is to say they're all alike. They all have digits 1, then 2, then 3, then 4, then 5. They always have the distal tips next to this area 3B, 3A border, and the wrist down here in that direction, right? The other way you can look at it, which is the way those guys looked at it, and they said, look how every one of these is different, right? Every individual monkey has a different looking map. These shaded parts are doing the hairy skin on the back. And you can see that they're not always in the same place, right? And you can also see that sometimes the representation here, number 4, is really big, where in another monkey it's be smaller, right? And so, since it was critical period, et cetera, one might think that sometime before the critical period was over, you had maybe some use-dependent differences, right? Or maybe it's just genetic, right? Monkeys they don't look different to us, but they must look different to each other, right? So their brain maps look different, so, what's the big deal? Well, then they thought, well, if it's the case that there's this critical period and it's all hardwired and it can't be changed, what do you suppose would happen if we amputated a single digit? Well, if you thought that, then this would just be quiet, right? There's no input anymore, so this part of the brain would do nothing, right? And so they did that experiment, and in my view, this is probably the best example of adult cortical plasticity experimentally that you'll ever see, right? So what they did is they mapped this monkey beforehand, then they amputated the digit, waited 2 months, and what they see is this part here, digit 3, which they had amputated, is this part of the cortex is now responding to the 2 digits on either side. So there was massive reorganization, right? This was an extremely unexpected finding, right? Because everybody knew that it didn't change in the adult, right? And here it changed quite dramatically. Okay, so that spawned a whole series of experiments in those 2 labs that we were just constantly trying to figure out what and how could you move these things around. Okay, so like I said before, all the distal tips, or the digits are always, or the receptive fields are always on one digit or the other. And so then he collaborated with this woman named Sharon Clark, who was a hand surgeon at Stanford. And what she did is she induced, and I think Mark talked about this a little bit, she induced experimental syndactyly, where you basically cut the skin along the fingers here, sew these parts together and these parts together, and you make a 4-fingered monkey. Okay? And these monkeys, they act like they're a 4-fingered monkey. I mean, they use these 2 digits together as though they're just normal. And then did the map. And what they found, this is with Terry Allard, they found that this whole huge region right here responded with receptive fields that crossed the suture line. Okay? And so this never happens in a normal monkey, right? And the thinking was, well, that's because now it's just like one big finger. And if the way you map these changes is, the way these maps occur is by how often do you get stimulated. And you don't get stimulated on both fingers very often unless they're connected. That's how they got separated, right? The cool thing that Sharon could do then, she always amazes us with her surgical skills. She could she basically cut the fingers apart and sewed them back up as they were before, and then went back and looked and said, are there still 2 receptive fields on both digits, which in fact there were. Okay, so it's not something weird like the primary afferent crossed the suture line or something like that. It truly was cortical. It wasn't peripheral in nature how they did this. Right? Okay, so if the idea is that if you have constant stimulation, right, or coincident stimulation, and that will change the synaptic weights on your cortical cells and change the receptive fields. And so then Bill Jenkins did an experiment that was also really cool. And he said, well, do you have to cut something off or sew something together? Can you just, by touching stuff, can you get changes in your cortex? And so he did that by teaching monkeys to stick their hand out of their cage onto this little aluminum disc that was spinning around. And on that disc, it had ridges cut into it. So the monkey had to reach out and touch this thing as it was spinning and get thousands and thousands and thousands of stimuli across its fingertip or two, right? And if it held it there for 10 or 15 seconds, then banana pellet would come out, and that was his only food. Okay? So basically, this monkey learned to hold on to this thing for food. Okay? And the reason it was important to rotate wasn't just so you could make the stimulus, but if you kind of ignored it, your hand would fall off. Okay, so you got to pay some attention to make sure that you're applying just enough pressure to make this work. So what's the results of that experiment? So here before, they did this differential stimulation, put their hand on the rotating disc, and here's the map after. So it was this little piece of skin that he videotaped that was always getting stimulated. And he said, well, how much of the cortex has a receptive field that includes any part of the skin? And it was this little gray part here. And then he said, how about after stimulation? And this is millions of stimuli that these animal experiences. And it becomes huge, right? There's this big growth here. And then the interesting thing that he also saw was that if you look at the 3A, 3B border that was defined physiologically, he didn't do any histology, but it seems pretty clear that that's where it was, it changed a lot, and that's shown down here, that there's a whole big region that now is cutaneous. Okay, it's not supposed to be cutaneous, it's supposed to just be deep. Okay, so this is about when I showed up to the lab. And this was about when your parents were making bad decisions that influenced your brain development and hadn't really met each other yet. But so I'm going to talk about this next. And so what I thought was, okay, we've been pussyfooting around this for long enough. You've been saying, Mike, that all this is related to your changes in perceptions and learning new skills and behavior, and you're not actually measuring any skills or behaviors. So why don't we do an experiment, sort of like Bill's, where you stimulate the bejesus out of a piece of skin, but you tell the monkey to tell you what it's perceiving, right? And if James and Skinner and all those guys are right, then they should get better the more they practice it, and that should change their brain, and it should be related. That's the way you've been saying it works, right? So let's see that. So that's what I did. And so this slide kills me. It's so ridiculously elaborate. Oh, I got a fiber optic light. You put a monkey in a box, okay? You tell it to stick its hand out and grab onto this hand mold, which is shown on the top here. And it was custom made for each monkey. And you teach the monkey to stick their hand on a certain way, so that's always the same. And there's holes in it for the different digits, and you can bring this thing up and you can stimulate the skin from underneath, right? So the trick is teach the monkey a trick, and then they'll get their food. What was the trick?
Is it tactile or?
It's tactile, yeah. So it's shown here. So this is how complicated I like to make things back when I was much younger, and so this is really the only important part. Okay? So what happens is the monkey puts their hand on the thing, and then the tactile probe goes up and presses against the skin, and then it vibrates at 20 hertz. And then it stays pressed against the skin, but doesn't vibrate. Then it goes at 20 hertz over and over until eventually it goes at something faster. When it goes faster, the monkey lets go, gets the banana pellet. If he doesn't, he gets a timeout. Okay, so it's that simple kind of thing. And I'll get back to this in a bit, but while he's doing that, there's a speaker over his head that's doing beeps that he can safely ignore. It's beep, 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 beep. Doesn't mean anything, right? He's got to focus on this, and when it goes faster, let go. So you teach the monkey, it takes forever to teach them to stick their hand out and do it, get it on right and all that stuff. And when you finally get to start doing the experiment, you see a function like this. This is the change in frequency, so this is 26 hertz instead of 20. This is the percent correct or performance or something. And by the third time he's actually acting like he gets it, he has a function that looks like this. Threshold is something around 8 hertz. Okay? So he's bopping along. And then what you do is, as time goes by, you make it a little bit harder, but he gets a little bit better. Okay, this is a couple weeks later, many weeks later, and very, very many weeks later. Okay, so he goes from being pretty crappy with an 8 hertz difference to now he's banging out 2 hertz trials pretty reliably. Okay? So this is exactly what's supposed to happen, right? They're supposed to get better, so now he can totally tell the difference between 20 and 23 hertz, whereas before we didn't even try it, right? So now he's much better.
Is that really the lowest resolution monkeys can get, 2 hertz, you think?
You'll see, there's the one that got just above 1 on average, right? Now the cool thing is, since I put those holes in different spots and the monkey understands the task, one day I'll sneak in there when he's not looking and I'll change it to the other finger. Okay, so now he does his unpracticed finger, and here you see that the untrained finger started pretty crappy and it never got better. Okay, if you just test it once or twice or 3 times over the course of months, it just doesn't change. Okay. So then what do? So you spend months and months and months training this animal to do this thing. And then you wait till he's really, really good. And then you get up the next morning, you anesthetize him, and you start to map him. And what we wanted to look at was both 3B, where we expected all kinds of cool things. We also wanted to look at 3A because Jenkins had shown that that border moved a little bit. So we thought, well, we should look there too. And Bill and I had an argument about that, a friendly argument. And he said that the reason that it changed is because he was doing the distal tips and it was just right there. And I would never see a change in 3A because I was stimulating the middle of the digit. Okay, it was just too far away. So we had a friendly bet on that. Okay, and then, so this is from the Journal of Neurophysiology paper. What you see on the left, every one of these points is a place where we stuck in an electrode. Okay? And I'm showing you an example from 2 animals that did the training, right? And then, so then once you get all that, you think, hoo-hoo, I'm done. But I was really anal compulsive in those days, and I said, you know what we ought to do? We ought to do the contralateral hand. That's a great control. It's unstimulated. It's in the same monkey, right? So then you flip the monkey over and do the other hand, 3A and 3B, and that's shown over here. Right? And so I'll just give you a flavor for what these experiments were. I designed it all double-blind and all that stuff. It was pretty lame to do that because if you look at these control hemispheres, and this is just the receptive fields, a smattering of what you got, and this is the size down here. This is what normal owl monkey 3B receptive fields look like. Okay, they're small and they're like this. Then you go to the experimental hemispheres where the skin was that was being stimulated, and they look completely different, right? So double blind meant nothing. You know, as soon as the guy maps this receptive field, he knows that this is a trained monkey and a trained finger and da-da-da-da-da. I mean, you just can't fool anybody, right? It's just so different, right? They have huge receptive fields, and they're really big. And this is where the monkey was stimulated, right here on this point. Okay, so that's a pretty cool finding. It doesn't really mean anything with respect to, you know, how did the monkey get better? He got bigger receptive fields. That didn't really seem to make much sense. But I also want to show you another control that we did. So remember these guys, these beeps that were happening. Well, I had another set of monkeys that were putting their hand on the stimulator thing, and they were feeling the exact same stimuli, but they were paying attention to these, and they were telling me when the frequencies changed, okay? And completely ignoring this tactile stimulus. Right? So they're getting the same thing down here, right? But they're paying attention to the sounds coming from up above. And if you map their hemispheres, they look like normal monkey hemispheres. Okay, even though they were extensively stimulating. So you had to pay attention for this thing to change your receptive fields in your maps. So like I was saying, receptive field size, I mean, it wasn't a spatial task. It was a temporal task. How fast is the thing vibrating, right? So in addition to just mapping the receptive field, we also took the stimulator gizmo and stuck it on that piece of skin, regardless of where the receptive field was, and asked what happens. Right? And the kinds of responses you get is either nothing, if the receptive field's on the thumb or something, nothing happens when you stimulate here, right? But if it is anywhere nearby, you can either get what I call the frequency following response, which is where you get a series of action potentials with every phase of the cycle, okay? And non-frequency, which was usually just an onset response and nothing to the rest, okay? So these are the ones that will tell the monkey, presumably, how fast the thing is vibrating. Right? And so we looked at and just asked a simple question, how many cells do this? And in the passive stimulation controls, so these are the guys that we were listening to the auditory stimuli, here for the stimulated digit, there was just right where they're supposed to be. If you were on the receptive field, you had a frequency following response. The dots are where you didn't get one, right? And the squares are where it was on the adjacent digit that, so the other side, right? And you see for both monkeys, this is all very well behaved. This is what normal 3B looks like. You don't respond to stuff unless your receptive field is right there. The experimental hemispheres look totally different. Okay, so what you're seeing here is stimulated digit, homologous digit, both digits. All of this area here in 3B, this is where it's supposed to be, and it has all this extra. Here it's supposed to be here, but it has all this extra. And look at 3A. 3A has a ton of frequency following tactile responses, okay, which you never see anywhere else. So, what to make of that? Okay, so what do you do with this? So you have these frequency following responses. Turns out there's all kinds of cool analysis tricks you can do. You can look at vector strength, you can look at interstimulus interval, and all this kind of stuff, interspike interval. But what I did is I said, well, you're trying to figure out what's the frequency of the stimulus, and is it slower or faster than the one before, right? So how could you look at this? In a simple-minded way, see, this is a good thing about being naive and not doing literature searches. You invent these really harebrained analyses. So what I did was I invented this harebrained analysis, which I called the two-cycle histogram. And so what you do is you divide this, so you take these two, and then you say what's the difference between this one and the next one, and this one and the next one, and this one and the next one, and you get some idea of the periodicity of that particular response, right? And then I did that by individual cells, and they all looked pretty much the same. And I thought, dude, there are so many of these cells in the stimulated skin, right? They must be doing something. What happens if you pool them all together? Because that's a signal coming out of the monkey's head, right? It's all the cells, not just that one. So I did that. I called it a population two-cycle histogram or something like that. And so what you see here is a trained digit. So I'm not showing the first cycle. That's over here somewhere. So this is really the difference from the last one, okay, is what you're seeing. And you see that the trained digit is super sharp and big, and the adjacent digit is broad and slower. Right? So what does this mean? Well, really the comparison is, what's the difference between 20 hertz and, say, 22 hertz? Right? Because that's where it's around threshold, or 28 hertz, or 30 hertz, or whatever. So that's shown here. And what you see is the trained digit, this is all trained digit, and the dark one is for the 20 hertz and all 4 panels, and the other one is for the other frequency, 21 or 22 or 24 or 26. So when you look at this and you think, okay, I've got some little monkey that's listening to all this out and deciding, should I let go in order to get my banana pellet, right? And so if you were that little monkey having to decide, this is all happening in time, not in space, right? And you can see there's a lot of overlap between these two, and it's going to be hard to tell them apart, right? Down here, this should be dead easy, right? This is super easy because they're not even overlapping in time at all, right? And these guys, these should be easy, those should be kind of hard, et cetera. So what you can do this for each monkey, for each digit that you got your data from, right? And then you can ask, well, what's the percent of overlap between these two? You can ask D prime, you can do all kinds of different metrics, right? We tried a whole bunch. And you can see that whatever the metric is, it should predict a pretty poor performance here and a very good performance here. Okay? And then you can say, well, what did the monkey really do, right? And compare those two directly. So that's shown on the next slide here. And this is for 2 different animals, right? And what you have is the behavioral, which I guess is open, and the predicted, which is closed. And you see for the trained and the untrained digit that they line up pretty well. Okay? And in fact, you can go even further and just take the threshold for that one versus the threshold for that one, the threshold for that one, and the threshold for that one, and do this for all your things and ask, how good is this simple metric of, I got sharper frequency following responses now, how well does that help me do this task? Right? And what you see is it works really well. Okay? So here you have the threshold that we measured in the monkey and the one we predicted from this population 2-cycle histogram thing, right? And some monkeys are, so here you go, Paul, this is it. This is the best monkey. It was 21 and a skosh, right? And then usually they're around 22 or 23. This one was really stupid, never really figured it out. Okay. But it makes the correlation really good. Okay. So what you get at the end of the day, I hated this monkey until it did that. Yeah. Okay, so we do a temporal task. We measure temporal response properties. And I should say we looked also at the cortical size and all that stuff. And it was correlated too, but much sloppier. This was the best correlation that we came up with. Right? And so, so there I am thinking that I did the right thing. And I was working with Christoph Schreiner then, and he was this big auditory guy. And he said, you're an idiot. You should look at the auditory cortex of these guys that your passive stimulation controls. And I was like, I'm afraid, it's auditory. And he says, no, I will help you. So he got me into the lab. And so remember, so we have this. And so a frequency discrimination in the auditory system, you can think of it, and some people do, and it's, I'm going to air quote the whole time. Imagine I'm air quoting everything that I say for the next few sentences. You can think about it as being a spatial task, because if you have one frequency, it's going to stimulate the hair cells at a certain point on the cochlea, and if you make it higher, then it goes towards the apex, right? So it's kind of like a spatial task. So in a sense, from the cochlear point of view. Okay, so we're going to do the spatial task in the auditory system. And so we've got a normal owl monkey. Christoph taught me how to do this. In the auditory cortex, it's a little bit out on the flat, and then it curls down into the sulcus. Okay, so that's why these are all straight, because they're going down the sulcus. But what you see is the classic isofrequency representations that you learn about in the textbook. And so all these guys are between 1 and 2 kilohertz, and these are between 2 and 4, 4 and 8, et cetera, et cetera. And so what I did then is said, well, let's look at all the points where the V-shaped tuning curve includes the frequencies that I tested the animal on. Okay? And that's these 4 points for 2.5 kilohertz, actually none for 5 kilohertz, and these 1, 2 for 8 kilohertz. And when you actually do this, you read in the textbook and you see the picture and everything, you have to do these octave bandwidths because it's pretty sloppy. It's pretty sloppy organization. Right? So that's why these aren't making a nice light line across them. Okay, what happens if you take an animal that supposedly used to look like this and teach it to discriminate these 2.5 kilohertz frequencies? That's what you get on this side. And sure enough, you get lots more points scattered all hither and yon that are responding to the 2.5 kilohertz stimulus. Okay, just like we would have predicted. Now keep in mind, this guy was the passive stimulation control. His 3B didn't show anything, right? Because he's listening to the beeps. Right? We looked at 5 kilohertz. This is a monkey that was doing the task and showed all huge 3B representation changes and 3A representation changes, and basically nothing happened to the sounds that it was ignoring, right? Whereas the monkey that was paying attention had a big representation change. Okay, so we did this for 4 different frequencies, and the last slide, the next slide, shows you this. So this is the trained frequency for the animal trained at that frequency, the animal trained for that frequency, and that 2 that were trained for this. These are the passive stimulation controls here, don't really do much. We had other just normal monkeys that we looked at, and pretty much if you train at a certain frequency, that frequency representation gets bigger. Okay? And you can do the correlation analysis and you see the same kind of thing as before. Okay? So what happens when you do something is you change your maps. Right? If you do a temporal task when it's vibrating your finger and you're paying attention to it, 3B and 3A go nuts. Right? If you're listening to a sound and trying to figure out what that sound is, your auditory cortex changes. Okay? But you have to be paying attention to it. Okay, I think I just said what I'm going to. So I didn't discover this. This has been hundreds of years old. Right? The temporally based task leads to temporal changes. The spatially based task leads to spatial changes. And you got to pay attention. And the other thing to keep in mind, which I'm going to come back to at the end, is everybody's different. Okay? They start at a different level and they end at different levels, and the representations change at different levels, but basically the same way. So there's a lot of individual variability. Okay? That we saw all that stuff.
Now, is there interaction effects now between the auditory and the tactile?
Are there interaction effects?
Yeah. Would you expect that?
Only if what you were doing made you pay attention to both.
Mm-hmm.
Right? So if you were doing some sort of cross-modal discrimination, then I would definitely predict that would happen. Yeah.
I don't know if you're going to get to this, so just ignore it otherwise. But if so, the human weasel binocular thing, you don't have to pay attention. You don't have to pay attention for ocular dominance homes for that to work. There's also the Jenny Saffron kind of statistical learning for auditory system, in which people are made not to pay attention, and it does work with babies. So.
So why does it happen here? You know, that's a good question, and I'm not going to get to it, right? Yeah. But let me just say that the thinking at the time was that you had to have attention and you had to have your acetylcholine and all that to happen. Mm-hmm. It might be more competitive than that. I mean, these monkeys were not only not paying attention, they were actively ignoring, right? Mm-hmm. So, I mean, that might have something to do.
That's sort of the case for like the Jenny Saffron. They're actively
I'm not.
Peak, you know, for the people who know they're trying to learn a grammatical rule by something saying peek up, pook up behind while they're doing some other task.
Yeah, that's a mystery to me too. Yeah.
But Greg, it's really necessary to call this attention, because in some sense, it's all about the contingencies of the task.
Yes.
Right. So actually, all you can really say is okay, how is a certain cue in the task coupled to the reward the animal gets?
Sure.
Right, so we don't even have to call this attention.
We don't have to.
It's just a contingency. It's coupled to the contingency.
Yes.
Wouldn't there be a safer way to interpret it? I'm just worried about you. You know.
It's not. It doesn't. If you use the word attention, you get more grant dollars though. So that's
good point.
That's kind of the way we floated it anyway.
All right.
Yeah.
So we agree.
Yeah, we agree. Yeah.
Let's call it consciousness then. That will.
Well, you know, that would be something to do too. Okay. So there I am finishing that, and thinking that these temporal tasks on the finger or frequency discriminations are pretty simple tasks, right? S1 and A1 are really nicely topographically organized, right? And the training alters the size of the representations, but not the general organization. And then what I was stuck in is I was thinking that interesting perceptions and behaviors don't really map on a topographic space very well. Okay? And after I left Mike's, I went to Bob Wirch's lab and I studied MT and MST in awake behaving macaque monkeys, because that was the optogenetics of its day, and I wanted to learn that technique. And MT, I kind of could wrap my head around. MT is a place that does visual motion perception, and it's linked to perception. Bill Newsom showed that really nicely. And it's all directional, and it's, you know what to put on the x-axis with firing rate on the y-axis. You get tuning and all that. I was hanging out a lot with the postdocs that were down in Bob Desimone's lab with Leslie Ungerleider, and they were doing V4 and IT and all that stuff. And it's like, guys, what are you doing? I mean, what's the difference between a red square and a green bar? And then what do you put on the x-axis? And a face, how's face being represented? How do you do that? And they said, you know, we still don't know, right? So it was like, how can you do that? And how can you study a simple percept that's not represented in the cortex in a topographic way? Okay, so that was the question that I had. And that brings me to this next topic here. Okay, so I'm thinking about this, and I'm going, how can you do this? And then I get a job, which was real serendipitous. If you know Mike Zaniga, sometimes you just end up with a job and you didn't even apply. You just shook his hand at a Christmas party, and the next thing you know, you're an assistant professor. And I was thinking, well, I got to do something now. And you know, I didn't want to go back to training owl monkeys and doing all the hard work and all that stuff. I liked using the macaque monkey, but I didn't want to do visual cortex either, because even in MT, I mean, I didn't want to do the IT and all that stuff because I didn't know what the heck was going on. And then at MT and MST, I had Tony Moshin and Bill Newsom and Bob Wirtz and all these heavy hitters, and it's like, what am I going to be able to do? But I had done that auditory thing before, and I was thinking about that, and I thought, you know what? Hardly anybody does awake behaving macaque monkey auditory stuff. There was Mort Mishkin and Joseph Rauschecker, and I think that was about it at the time. And I thought, well, if I did that, I could certainly break into the top 5, right? I mean, I got a shot at it. And so then, well, okay, what am I going to do? And then I thought, you know what? I got this poser here. How can you do this? How can I do this? And then I realized sound localization might be the way that you could study this in a cortical area, right? Because the lesions of the auditory cortex result in sound localization deficits, okay? But only in the contralateral hemisphere. So you need your cortex to do this, okay? So at least it was cortically mediated. There's no topographic representation of auditory space in the cortex, just not. A lot of people have looked for it. Nobody's got any evidence that there is anything like that. And the key to sound localization, as opposed to V4 and IT, is that we know what the nervous system is trying to represent, azimuth and elevation. So I know what to put on my x-axis, right? So I decided, okay, I'm going to work on monkeys. This is a macaque monkey. If you haven't seen one before, they're, you know, look very intelligent, and they're very introspective and learned. They've made we know a lot about the visual system because of these animals. These are super smart animals. These are the ones that the visual guys get. The auditory guys were still new, so they give us these ones. But I'm used to working with dumb monkeys because they got caught in the wild, so this seems about appropriate. Right? So if you take everything unimportant away from a rhesus monkey, this is what you're left with. And what you have here is the lateral sulcus and the central sulcus. So 3B is buried down in here. A1 and auditory cortex is buried down here and kind of comes out on the end like this. Okay, so what we do is we anesthetize the animal, put on a recording chamber, let them wake up, teach them a task, right? And then every day we can take an electrode and we can stick it into the brain through some unimportant parietal lobe or something, and then record from the superior plane and the superior temporal gyrus, right? So we take metal electrodes, we stick them up close to cells, and we record the spikes, okay? So all the things that I'm going to talk about for the rest of this time, there's no laser, there's no transfecting anything, there's no transgenic anything. I'm taking a stick and putting it in a brain and counting the spikes, okay, and trying to figure out, you know, when did they happen. Okay, so that's what we're going to do. We're going to just count spikes that we got from sticks. Okay. If you look down on the auditory cortex, the way it's organized is that there's the so-called core. So this is A1. All animals have an A1. And the primate that extends to this core field, there's 3 different fields. They're surrounded by these belt fields, and then there's this parabelt. And this is all in the sulcus here, and this is the part that kind of comes out. And Joseph Rauschecker, when he was working with Mort, thought, wouldn't it be cool if we have dual processing, what and where, where and what pathways, just like in the visual cortex, and like many before and after him, let's just stick visual cortex rules onto the auditory cortex and see what happens. And so the idea here is that processing starts in A1, goes to CL, and that's spatial processing, and then it goes this way for whatever non-spatial processing is. Okay? And so what I did is I stuck my recording cylinder right over here, and I recorded from all these different cortical areas and asked, how well can the monkey localize sounds, and how does it match up, right? And the prediction is it's going to work really well over here, it's not going to work so well over here, or whatever. Okay, so what did I do? So I thought, I'm going to do it just in azimuth for these experiments, and I'm going to change the intensity of the white broadband noise. And the reason I chose that is because different neurons respond very differently to different amplitudes. It's not like a contrast sensitivity function in the visual system. They don't all get more spikes the louder you get. Some go up and down, and some start high, some go flat, right? So there's lots of variation, but your sound localization ability is really stable over a broad intensity range. Okay, so now I'm going to have different neural activity and sort of the same behavior, and so maybe that will give me some insights how to do this. So what happens in practice? So the monkey sits in a sound booth with this ring of speakers around it, and it's trained to listen to these sounds, and they're 200 millisecond broadband noises, so they sound like psst, like that. And they'll come from one speaker, and then maybe louder from another, and then maybe another. And when it comes twice in a row, basically a one-back task, you let go of the lever, you get some juice. Okay? And you do that for an hour. At the end of the hour, I can count up the spikes. Okay? So what you're seeing here, each one of these raster plots, each line is a single trial, each dot is an action potential, and they're all sorted now by the speaker location. They came out randomly to the monkey. Right? Now, you don't have to be a neurophysiologist to go, hey, Greg, there's more dots over here than over here. Right? So this is the tuning. Okay? You also don't have to be a brain scientist to go, you know, there's dots on all of these. Right? So the tuning is really broad, but there's more dots over here than over here. Okay? And then if you look at the individual neurons, right, and instead of plotting them like this, what I'm going to do is take the average for each of these sets, and I'm going to plot it as a PSTH. Right? And here are, 16 cells, just to show you the variability. There's 4 lines in each of these, which corresponds to different intensities. So this one is very well behaved. It's lots of spikes, and this is the peak response. Lots of spikes in the contralateral hemisphere, when it's loud, less when it's quiet, hardly any when it's quieter still. Right? Here's one that kind of breaks the rules. It's the best response at 35 dB. So I should say 25 dB, which is the quietest, is quiet. Right? And 75 dB, which was the loudest, is pretty loud. That's about, so you guys are hearing me at about 70 dB or something like that. Okay. But this stupid cell fires way better to the quiet sounds here than to the loud sounds. This one has 2 bumps, and they're all wacky. Right? So what do you do with this kind of data? What I did was everything that everybody did before in the cat or whatever, just to see what it was like in the monkey. First thing you can look at is where's the best direction, which is just the direction that goes straight up. And what you see for all this, for A1, CL, CM, ML, MM, and R, so this is supposed to be the space guys over here, and these are the non-space guys. And there's a pretty even distribution across, and they're not significantly different from each other. And you'll notice that sometimes they're ipsilateral fields, but mostly they're contralateral. Okay? And we looked at a couple of other metrics. So one was this location index, which is also known as the Baker index, which John Allman came up with for MT cells, probably 20 years before I did this. And it's 1 minus the response in the worst direction divided by the response in the best direction. So in this particular cell, if you see here, if you take the red line, the best direction is pretty high, the worst direction is pretty low, and you get something that's close to 1. If you look at the blue line, the best is this much, the worst isn't much different, and you get a little number. Okay? So the closer to 1 you get, the more sharply tuned you are. Right? We looked across all the population of cells. There really wasn't much to do with the different intensities, but what you see is here's A1 here, CL piles things up towards 1. Okay, so it's sharper, right, as predicted. And these other ones are not, right? And finally, we looked at bandwidth, and that's just you go to the half height, right, and see how far you go. So it's going to be 230 degrees in this cell for the quiet sound, and half height here is all of them, 360, right? This was pretty common if you look at the same kind of thing. So now what you're seeing is each individual animal for each different intensity. CL is the lowest. The rest are pretty high. Right? And these are medians in first and third quartiles, so there's lots of variability. Right? Okay, so if you're looking at this, you're going, okay, Greg proved it. It's a CL. That's the space place. We're done. Right? But remember, I'm trying to figure out how is it that the monkey can use this information to tell where the sound is and see that it came from the same place twice in a row. Right? So how do they do that? And when you think about this, look at this. This is 90 degrees. Okay? 90 degrees. That's this much. Right? So that's pretty darn big, right? And so the question is the spatial tuning good enough to account for sound localization ability? And what's tough is, the way that I designed this experiment, is I really don't know what is good enough, right? Now I can take a monkey and I could train it to tell me which speaker it thought it came from. Remember, I just had to say it was the same place twice, right? I can teach a monkey to do that. You can't do it when their head's fixed and they're sitting there because, you know, it's somewhere back there. How do you, monkeys can't point for squat, right? They're really inaccurate. So I thought, well, the way people do it in ferrets and cats and stuff is they have the animal freely moving and it starts in a spot and then you play the sound and it goes to the speaker and gets a drop of juice. If I did that, I put the monkey in there, he's going to rip my speakers and tear the foam off the walls and have a great time and not get anything done, right? So I thought I should use a different species that has very similar sound localization ability. And I had done that before, and I had a good species in mind because they don't tend to trash the place, and they do tend to give you that kind of information. And that's what I like to call the naive Aggie. Okay? So you get yourself some naive Aggies. And naive Aggies are very much like monkeys in a lot of ways. They vary in their intelligence level and stuff. The smart ones tend not to do psychophysical experiments, right? They go do vision experiments or something. So I end up recording from this guy, and guys like him, right? But you can tell them what to do. And what they basically had to do was listen to they sit in the same booth with the same speakers, heard exactly the same sounds, and I had a little clipboard and said, when you hear the sound, tell me which speaker you thought it came from, right? So they could identify each individual one. Okay. So you hose a guy like this off and give him $15, and he does that. And what you end up with is this. So this is the error as a function of location for these 4 different stimuli. And pretty much everybody except the dark blue one, which was the quietest, you do pretty well. Okay. So like I said before, there's not a whole lot of variability across intensity ranges, and there's a little bit when it's kind of quiet. Okay. So that's what I have to figure out, how to do this sort of performance with this kind of data, right? How are you going to do that? Well, you're going to do it by trying everything that everybody else has done before, right? And so that's what I did. I used an ideal observer probability model, which has been in the literature for a long time, a linear pattern discriminator, population vector, et cetera. And I did all those, and what bugged me about all those 3, anyway, is I do it, and they all kind of make you set a threshold to decide, you know, when am I going to call it that one or this one or whatever. And I could manipulate my threshold, and I could make it so A1 was the best space place. I could make it that MM was the best space place. I could make it CL was the best space place. And it was like, who am I to decide how to do this, right? This is dumb. And then I knew about this. This is a logarithmic maximum likelihood estimator that Tony Mawson had come up with in his group, and that did direction of motion in MT, which is the same problem, right? Because I have 360 degrees, and I have to do essentially the same thing. But it was really complicated. So I recruited Lee Miller, who's my colleague at Center for Mind and Brain, and he MATLAB'd it and came up with it in just a few minutes. And basically how this works is you imagine that, here's the data that I showed you before. But the monkey doesn't have access to all this, right? The sound plays, you have to decide, right? You don't take averages and all that kind of stuff. So what Lee did, is I gave him all my data, and he said, what we're going to do is we're going to take one trial, from one neuron, say this trial right here, that one, and we're going to imagine that the little monkey inside the monkey who decides where it came from has knowledge, has general knowledge about the tuning of each of these different units, but it only gets 12 spikes, okay? And it's going to say, what's the probability that the sound came from here, given that this cell gave me 12 spikes, okay? And then it'll be some probability. And then I'll do that for another cell. And that cell will probably have a best direction someplace else, right? But I'll say, what's the probability that it came from here? What's the probability that it came here? And I'll do that to 100 neurons. And at the end of the day, I'll look and see who is the loudest, who says the most. That's the one I pick. Greg doesn't pick anything, right? I'm taken out of the equation entirely, right? Then you can imagine on another trial, maybe from this neuron, I'll take this one. And I'll say, okay, I got 3 spikes. What's the probability it came from here, given that you said 3 spikes? Now, it's very low, okay? And some other neuron has the best direction here, and it's going to be very high, so it's going to win, right? So you do that, and we did it for contralateral space and ipsilateral space. So in ipsilateral space, you get horrible estimates, right? And due to some tweaks that only Lee Miller can describe, they're actually worse than chance, right? And then we looked across the population in contralateral space, and what we saw was these data here. So this is MM, which nobody knows what it does, and it wasn't very good. ML is supposed to be a what place. R is supposed to be a what place. Here's the human average with the standard deviation. Here's CL. CM almost makes it, and CL is dead on, right? So basically what this is telling me is that if you look across the population of A1 neuron, of CL neurons, right, and you have some general idea about the tuning properties of these cells, you can have this completely non-topographically represented region tell you with high fidelity where that speaker came from, okay? Where that sound came from, which speaker. Okay? Which I thought was really cool because that's probably the way that a lot of really cool things that we use our brains for, that's probably how it's encoded, right? It's encoded across this population because you can't topographically represent everything. Okay. Okay, so that was cool. And then I moved into this category, which is really depressing. And I realized that you guys are young. This is awesome for you guys, right? So I thought, what happens when you get older? Because all the older people that I knew when I was young would complain about, oh, Greg, you should study aging because hearing sucks when you're older. And it's like, it's for old people, right? So now I'm really enthusiastic about it. So young, you guys are like this. These guys are cool. They're strong. They rock. They're awesome, right? That's what you want to be. You want to stay young, right? You can't really stay young. Okay, so then you end up being middle-aged, and maybe you be governor or something, I don't know. Or you still rock out, and that's fine. And then inevitably, you get old, okay? And you don't really want to be old, but it's better than being dead, right? So there we are. And if you get, a good hair dye thing, you don't look that old after all. And I don't know, let's not even talk about Keith Richards. Okay. So he's like 200 or something. So what happens when you get older with respect to hearing? Well, it goes south. Okay, so age-related hearing loss or hearing deficits is probably a better way to put it. It's a major health concern. It's the third most common chronic condition in the aged. It's behind heart disease and arthritis. Okay, so almost everybody gets it. These are really conservative estimates. These guys are coming up with worse and worse numbers for people as we go. People are getting older and older and older, right? So this is going to be a major deal as we get really old. So what happens? What is the complaints, right? Very rarely does a person come in and say, hey, doc, I got age-related hearing loss. They don't say that. What do they say? The major complaint is a reduced ability to understand speech, especially in a noisy environment. Now, I don't know who these people are. I pulled this off the internet, but this really shows what happens to these people with age-related hearing deficits. So here you have grandma, and she's making hot dogs with Junior here, and they're all happy, and there's mom looking lovingly down, and there's the teenage girl embracing her father in all good-natured way. We've all experienced that, guys. And here's grandpa, not really keeping up. Kind of looking like,
Oh, he's just fed up with her nagging.
Look at him, he's just going, geez, don't drop my hot dog. I can't hear a damn word anybody's saying, right? Okay, so what does that mean to a neuroscientist, right? People do, actually, usually it's not him that says, hey, doc, I can't understand speech in noisy environments. It's these two will say, we got to get this guy a hearing aid or something. He's driving us crazy. Okay, so what happens in the lab? What do we call that, right? So if you take these older people in the lab, you find that they have two primary deficits. One is in the ability... It's my laboratory. I always wanted a big switch. I think I have to get one, just hook it up. One thing that happens is it's really hard to localize sounds, okay? And the other is it's hard to follow fast temporal things, the envelope of the speech. Those are the two main things that you can do when you're impoverished stimuli, et cetera. And that makes sense, because if you're in a cocktail party, like we're going to be this evening, right, it's really hard to hear people, but we can localize the person we want to listen to pretty well, and we can follow the envelope of their speech, which is what the cochlear implant does. It basically just does the envelope. Right? Okay, and if you're old and you can't do those things, it's really hard. So here's a tip. If you're ever talking to an old person who has age-related hearing loss, get in their face, and you don't have to talk that much louder, because this happens to people even if they have normal audiograms, right? Just talk more slowly, right? And it'll be a lot easier. Okay, so can we do this in monkeys? Well, we can, because the Primate Center, which is just up the road from campus, or from our labs, has thousands of young animals and scores of old animals. In monkeys, macaque monkeys, it's said, I guess, age about 3 times to that of the human. So a 25-year-old monkey is like a 75-year-old human. Okay? The oldest monkey that I've ever seen in the literature was the equivalent of 107 years old or something like that. Okay, so then they usually die in around 30, right, which is around 90. Okay, and they get the best care anywhere. They have a bunch of vets, et cetera. So. They're not like wild monkeys that get caught by the line because they're slow, right? So they get to get nice and old. Now, if you look in rodents or monkeys or people to a lesser extent, what you find is you get the age-related hearing loss, these deficits, even if you have normal audiograms. But what happens is you get all kinds of bizarre changes all throughout your brainstem and thalamus. And it's believed, mostly from Donald Caspari's work and others, that what's happening is there's reduced excitatory drive out of the cochlea, even if you have normal hearing. And what happens is that the brainstem and the midbrain and the thalamus are trying to adapt for this by decreasing inhibition. Okay, so there's all kinds of evidence that the general conclusion is that the brain is trying to compensate by decreasing inhibition throughout the way. Okay? And different species do it in different ways, and different places do it in different ways with calbindin or palvalbumin or the expression of this molecule or that or whatever. Right? So what I'm going to do is the same experiment I had already showed you guys in these animals, and I'm going to do it in some of these guys. Okay, so here's the audiograms. These are behavior. I can teach a monkey to press down on a lever. When you hear a sound, let go, and then you just make them quieter and quieter. And this is the threshold at these different frequencies. The two old monkeys are dark, and the two young monkeys are light. So these are 77 and 79 versus 31 and 43. And importantly, these are the noise stimuli that we're going to use. Okay, so these monkeys can hear fine. Okay, so it's not, you don't have to compensate for a hearing loss. And they did essentially the same task, and we counted up the same kinds of spikes like we were doing before. And so now the question is, let's see what the responses are. Okay, so here's the old monkeys, and here's young monkeys. This is spontaneous activity, so this is just right before the sound is going to come on. And these are medians and first and third quartiles, and sure enough, there's higher spontaneous activity in the old monkeys, which makes sense because they have this reduced inhibition. That's what we were taught, right? Compared to young. This is an area CL. We just did A1 and CL because that's what we just did. And in fact, these are statistically significantly different than these. So there's more activity. Okay. If you look at the young guys at these different stimulus intensities, and then you see that it gets a little bit bigger as you go, right? But not a great deal. And the question is, what happens to the old monkeys? Well, I would have preferred to do all four of these, but we couldn't because they don't work that long. They get tired, they fall asleep, etc. So we had to pick one, and after much debate, we decided to go with 65 dB because we didn't really know how well they would be able to hear at the end of the study, which is when we took the audiograms. And we figured, well, we'll be in the ballpark here. So let's look at 65 dB because it's the only one we did, and it's off the charts. Okay, again, these are medians and first and third quartiles. There's essentially no overlap between these populations. Okay, so they have higher spontaneous activity and super loud driven activity. And I can walk into the lab and just by hearing the spikes know whether it's an old or young animal that's doing the task, right? Because they just do way more. Okay. Okay. So how's their spatial tuning? Yeah.
Question.
Yeah.
Isn't a response in the range of 5 hertz extremely low for a cortical area that's driven by a tone?
Yeah. I've done this a whole bunch of different ways. These are the average across all locations. Okay. So I'm doing the lousy parts too.
Okay. So this is really the mean over the whole area.
Of the yeah.
Okay,
yeah.
so it might, okay.
Thanks for pointing that out.
What's the frequency used for the testing tone?
The frequency? It was a broadband noise.
Okay.
Yeah.
White noise.
White noise.
Okay.
Yeah. Okay. So we have a bunch of these. And then I should say that Dina Wara-Salinas, who's now in Alan Basbaum's lab, did a lot of this work, and so did James Engel, who was a postdoc in my lab at the time. And what James hated these, and he likes technology and stuff, and he said we should boil this down into a single figure so that we can look across the population. And so what he did was he took these same data and he'd say, okay, this is the best direction, I'm calling you 0, and this is minus 22.5, minus 45, plus 22.5, plus 45, and just do it like that. And I'll do that to every cell, so everybody lines up on their best and then flanks out around. Right? And then I can do a color plot as a function of time and see what's the output of A1 as a function of time, in that general way. So this is what you get. This is in young monkeys. And so here's the location. This is to the right and to the left. This is when the stimulus is on. Okay? And red is lots of activity and blue is no activity. And you see everybody lines up at 0 like they're supposed to. And you can see it's pretty broad. I showed you that before, right? So the best activity is on the order of plus or minus 80 degrees or so, but it responds to everything. And then there's a little tail of a response and a little offset, and then you're done. Okay? And you can do the same thing for CL. and you see, ooh, neato, they're sharper. I did belabored that last time, showing you all these fancy metrics, and here he comes and goes, look, it's sharper, right? So it's sharper. What happens when you do these to old monkeys? Okay, these are what you get with the old monkeys, and you see they're different. Okay, and they're different in a bunch of kinds of ways. One way is that, well, the A1 kind of looks the same. It's really broad, right? Doesn't really last quite as long. There's not much of this tail, but there you go. And then CL looks way different. It doesn't get sharper. Okay, it stays very broad. And the other thing that's probably hard for you guys to see is that the latency is even and shorter, and I'll show you that a little bit better in a second. And then there's this business, right? What's this oscillatory behavior? That's new too, right? Everybody who's ever looked at this and thought about it gets this kind of expression like, right, including me, right? And I still don't, if you don't have that expression, if you go, I would really like to talk to you. Because I don't understand this.
You have normalized this now, right?
Oh, absolutely.
And
if I had,
peak, and the peak values are rather dramatically different.
Very dramatically
right?
different.
So
yeah.
maybe the tail there for A1 might be comparable in its response amplitude, but you just won't see it because you normalize it against a much higher.
That might be true because if you use this value over here, right. this is all blue. Mm-hmm. Yeah, you can't see anything. Yeah. Okay, but the latency thing I can wrap my head around. Okay, so what James did next is said, this is all cool, all these different colors. Let's do it a little bit differently. Let's just look at the first 50 milliseconds. Okay, so we'll get rid of all this stuff, right? I'm going to take the first 5 milliseconds when the sound is still in the air and it's not counting yet. I'm going to count the spikes. That's spontaneous. I'm coloring that green. Okay. Then I'm going to ask for each of these, pixels, if you will, are you above or below that statistically? Right? And if you're below it, you're blue. If you're above it, you're red. Okay. And let's see what that looks like. Okay. And this is what it looks like for the young guys. And this is pretty cool, actually. So you see in A1, there's a little bit of blue, right? And pretty even latency across location. And in CL, you see lots more blue that's kind of carving out this response. So it's carving out the flanks. And this is consistent with all kinds of stuff that we know, that some sort of suppression or inhibition, lateral inhibition or whatever, is basically forming a sharper spatial tuning curve than you normally get. Okay? And you get the longer latency because CL is a second order place. Everything makes perfect sense. Right? Then you do it in the old monkeys, and you see something completely different again. Okay? So was there an audible gasp? Did I hear it? It's flat. There's no blue. Okay, that's the first obvious thing to see. It's straight, right? It's down here around, 12 milliseconds, whereas these guys are around 20, right? So it's super short, and there's no difference between the two. Okay? This is completely consistent with this high spontaneous activity, right, and lack of inhibition. Okay? And it's telling me that what's happening here is that whatever is doing this is gone for these older animals. Okay? And that's what's screwing the whole thing up. Okay, and for some reason I have this on here again. I'm not really sure why. I think it's to make the point that A1 and CL, except for the amplitude, aren't a whole lot different. Okay, because that's what I say here. It's about the same. And there's not a sharpening in the spatial tuning from A1 to CL.
Oh, wait, why do you say it's the same? It look rather different if you go back.
Yeah, it is a... Sorry, this part is, right?
Which
yeah.
part? Here.
Well, the spatial, so this is showing us the spatial tuning, right? This is very different. The latency is very different. Yeah, it's completely different. Okay. I should probably spatial tuning. Yeah. Okay, and what you see is there's not a sharpening of spatial tuning from A1 to CL in the old monkeys compared to the younger animals, and I think this is scary, right? Because if you think about how we like to think about how the cerebral cortex works, we think of it as you come into the primary area, and then in these subsequent stages, you carve out different features, and that's how you represent different things, and that's why we're so cool, right? And if you can't do that very well, what happens to things that take a lot of processing to get to? Like cognition, right? So being poorly able to move along the hierarchy could be a very general mechanism of general cognitive decline. That's why as you get older, you don't necessarily have Alzheimer's or anything, but you're just moving slower, right? And that could be part of the way. Okay, so, yes?
Yeah, just on that, but there's a bit of hope in the visual system.
Hope, you said?
Hope.
Oh.
Hope. bring your agent ears. Okay. Yeah, okay.
Talk slowly.
Okay.
Look right at him.
All right. In visual system stuff, looking at agent perception, there's a lot of things to the effect of, say, if you look at isolated letters, giant age effect. But if you look at same letters in text, no decrement whatsoever. So maybe, and the interpretation in that literature is that you go much more into a sort of top-down inference. And
I'm not familiar with that literature.
And you bring more cognition to it. So maybe you might, if your periphery is getting bad, you might want to flatten that periphery out and bring all your inference to the...
Yeah, that might be a way to do it. Yeah.
From a cognitive perspective, disinhibition also has its advantages.
From a cognitive point of view, vision
Yeah.
has its advantages.
Yeah. You're less norm-bound, you know.
Yeah.
That's why we drink alcohol, no?
Okay. Are there any other questions? Because I'm going to get... Yeah.
Is there any effect on, let's say, early training? For example, if you train young monkeys to recognize like maybe a sharpened tuning curve, and when they get old, do you see this effect again? Like a decline
Okay,
in
unknown, right? In monkeys, certainly. So there's, I used to be, I got into the auditory thing to be in the top 5, right? As far as aging in the cortex in monkeys goes, there's me and a guy in Utah, right? So I'm still in the top 5 there. Yeah. And if you do auditory, I'm the guy, right? But there's been work in rats, and what you can see is that you can do, you see the exact same kinds of things, right? They haven't done space yet, but they've done temporal processing things. And what you can do is you can train them up with luminosity kinds of things, and they get better, right? And they have differential gene expression, and they do all that kind of stuff too, right? So, but I don't know, no one's ever, I mean, I'm the one who's done anything, so, and I didn't do what you suggested, so.
So we're screwed.
Yeah, we're, for now,
Yeah.
yeah. Top down, okay. Okay, so what, if anything, does any of this have to do with the development and or with evolution? Okay, so now this is purely speculative, and you can fall asleep or leave or stand up and storm out or whatever you want, because I'm just going to make stuff up now. Okay, so when you think about the somatosensory cortex, I'm going to go back to that. You're used to the homunculi, right? And what I showed you is that you can mess with this homunculus a little bit. Right? You can make its finger bigger or smaller or whatever. And that's the somatosensory cortex. So I didn't have the courage to ask Leah for a slide, so I just took it off the internet. But I like this one because it's a circle. And it's showing the primary somatosensory cortex, which is in red. And here it is in the macaques and the marmoset, which is a New World monkey as well. And the point here is that it's true that there's somatosensor S1, if you will, in all these different mammals. It's a little bit different in primates, though. Okay, so and there's been some debate. So in primates, it's considered to be area 3B, right? So this is taken from Jeff Padberg's paper. He was a postdoc with Leah, and it shows really nicely that the somatosensory cortex of the macaque, you have 3B, which is S1. It's this cutaneous representation, and then there's 3A, which we talked about, which is deep, right? They call it deep, and then area 1 is also cutaneous, and they have opposite maps. So the digit tips are over here, and then it goes to the palm, and then the palm and the digit tips. Okay? And then there's area 2, which is also deep. And then you go to area 5, which is deep and visual and all kinds of cool parietal lobe stuff. Okay? So this is how it looks in the Old World monkey, in the macaque. Jeff also did a really cool study that I've been chewing on for 10 years to try to figure out what it really means. And here again is the macaque. This is from Tim Pons. This is a study that Liz Disbrow did with Leah. And here you see area 2, which is deep. There's this cutaneous little bit here. And then you go into area 5, which is visual and deep. And then Jeff did this in TD monkeys, I think, right? And what you see here is that there's 3B, like you're supposed to be. There's 1, which is cutaneous, like it's supposed to be. And there's really not really much in the way of an area 2. It kind of goes right to area 5, right? So do New World monkeys have an area 2? It doesn't look like these particular ones do. I did a little bit of homework. I remember Kelly Huffman, when she was in Leah's lab, did a bunch of mapping of 3A, which is up here, and you guys called it 1/2 because it's just like kind of weird, right? So is there a 2? So how would you make a new cortical field, right? How could you actually pull this off? Well, and we learned a lot in the last few days about that. Okay, so one thing is, well, you need some sort of substrate for this sort of thing, right? And if you look, and this is a paper from John Costa's group, if you stick a tracer into, say, 3B, and you look and see what are all the neurons that project to it, you see that they're all over the place. And if you do neuroanatomical studies and you inject tracers like Leah does all the time, you see patterns and stuff like this. It's not from one box to the next. It's all scattered all over the place. Here I'd like to give a shout out to Jimmy Dooley. This is from his thesis. He was a graduate student of Leah's until last week when he just finished his dissertation. And this is an injection into the rostral part, just rostral to S1 and the monodelphus, and you see that it's getting input from all kinds of different places. Okay? And these are generally also reciprocal, right? So it's getting input from a bunch of places and it's sending output to a bunch of places. Similar deal, this is again from John's group, and you see patchiness all over the place. So my point there is that you have all kinds of widespread anatomical connections by which you can mess things up with, right? And then remember these guys, right? So we train up some animals, and they get better with practice, and what you see is that you get this nice correlation, right? Now what I want to point out here, which gets to Paul's question a little bit too, is this is from 4 different monkeys, and these are the learning curves of these 4 monkeys. Okay? So some monkeys, so the dark ones are the trained digit, and they start out, and over the course of a month or 2, you get better and better, except for this guy, right? And the untrained digit doesn't really get much better, and that gives you this enormous variability. Okay? So different animals can learn better things differently. Right?
How do we see here that the untrained digit does it get better?
It doesn't get much better.
Now, where do we
this
see
one does. This one did. That one didn't. That one technically did. That one did. This one didn't. Right. So it gets a little better, but it stays bad. Okay.
It was actually interesting that the non-trained digit would also get better.
Yeah, but remember they had the really big receptive fields, and a bunch of stuff happened there too, right? So right. And it's on this thing, and so it's getting some kind of stimulation, and if it can feel anything, it's going to use it all. Right? Okay. So the kicker here is remember these. Okay. So here I'm showing you the area, just 3A, so 3B's down here. This is 3A, and now if it's a dot, it's a well-behaved 3A cell that's deep. And if it's colored in, it's some sort of cutaneous response. Okay? And what you see, so here's the other hemisphere of 3 of these guys. This is all pretty much normal. This is the passively stimulated. This is all pretty much normal. But some of these guys have huge amounts of cutaneous receptive fields, right? And some have less, right? So these guys are the ones that did better, and these guys are the ones that didn't do as well. Okay? So you have enormous variability also in how much your brain changes with this. And originally, when I thought about this, I thought, okay, this is fine. This is very adaptable because if you can change your brain, right, you're going to want to change your brain because the seasons change, right? So if you're an insectivore or something like that, or if you like to eat bugs, you're not going to get many bugs in the wintertime, but you will in the summertime, so you have to change your strategies, do different things. You're not going to use your hands in different ways depending on the season. I mean, that makes sense. And if you can move your brain maps around, you get really good at it, and you think, oh, yeah, first day of snow skiing, I'm not that good. Last day of skiing, I'm much better, right? And so I can get good or bad or whatever. And there's precedence for animals changing things by the seasons. They can change their fur color. They can change all kinds of stuff. Okay, so I happily bopped along for, until Jeff's paper, thinking, this is all that there is to it. There's nothing to do. It's got nothing to do with evolution at all. Right? And then, of course, what happens, though, and Barbara brought this up, what happens if it hits the fan, right? When the meteor hits, right, even if it's a local event like in Siberia, if you're used to live around here, you're going to have to move or change your strategies tomorrow, right? If it hits, the Earth when the dinosaurs are around, you have to change your strategy today, right? Because everything's going to be different for probably the rest of my life. Or if it goes more gradual, you might have to change your strategy too, right? So if you have to change your strategy, right, who's going to do better, right? So this is a monkey that basically, if you didn't know anything, right, you walked along, you would not call this area 3A. This is a completely different cortical field. This monkey made a cortical field in 2 months, right? Of just, I got to listen to this so I can eat, right? And I got good at it, and I made a new cortical field. Because whatever I need to do, okay. So what did 3A usually do in this guy? I mean, what was the deficit? That's something Mike Zaniga always asked me. It's a zero-sum game. You change one thing, you have to lose another, right? And that's certainly when, the precursors and what Barbara was talking about on the owl monkey, how you changed your fates and you delayed development and you increase it or whatever, you make more neurons, and now you can do what 3A used to be able to do, or now you can move it over and do something new, et cetera. So that's what I mean by evolution on a nanosecond scale, right? You don't need 60 million years to make a cortical field. I made one in 2 months, right? And if you do this kind of stuff, and then it hits the fan, and now you have to, eat different kinds of bugs or whatever, then these are the guys that are going to be able to move forward and procreate, right? And hope to get new gene expression and all this other stuff that we heard about up to now. Okay, so that's my hypothesis on how you can go from this to that, right? And it doesn't necessarily have to take a super long time. Okay, I think I will finish there. I'd like to thank everybody that participated in this work and in my thinking. Certainly everybody who paid it for it, but mostly for you guys for sticking with it and holding off lunch for a little while. So thank you all very much.
Great. I think. What? Okay, young people first. Of course, they're all stacked at the other side of the room.
Hi. Thank you for the talk. It was really, really nice. I just, when you talked about how the degradation of hearing when you get older, I just kept thinking of my grandmother, and I was wondering how you could explain the fact that she's like 93 years old. She barely hears anything. Like we're like, "Hey, grandma, you're a pill" or something. She never hears it, and then suddenly, when you start mumbling or you say, "Oh, we didn't hear dessert," she goes like, "I heard that." Is there any sort of selective hearing in the elderly or something? Because I really cannot understand or explain it.
Well, there's a long history of selective auditory attention and listening, but as a husband, I'm not allowed to divulge that secret. Otherwise, I'll get kicked out of the club. But another real hallmark deficit in aging is attention-based, right? And it has been shown to fluctuate. So they have a harder time with directed attention, and it can wax and wane. And so one plausible explanation, other than that your grandma she's wise, right? Certainly, right? And so there's always kind of a good idea to keep in mind what's going on and just pick your spots. But it could be that sort of thing too, where it's just hard to pay attention, and so that's when it's harder to hear.
No, I think your grandmother just figured out how gullible you are, that's all.
Thank you for your talk. It's very interesting. We have been working with sounds for many times here, and I have two questions. One goes in the same direction as the previous questions, that if you have tried to, because you're talking about the cognitive load of the sound location. So that means if you're talking about cognitive load, it's much concern, you have to concern about timbre. Because, for example, if you're using wide spectrum, white noise, you don't convey timbre information. And for many aging people, timbre takes a lot of importance because they know by memory what the sound means. Mm-hmm. So that means, this is my first question, it has been done some research taking care of the location and aging connected to timbre and memory. That's the first one. The second one is, that's binaural location is based very much in the position of your head, in relation to the sound source. It has been ensured that the location of the head of the monks or the other guys are really in center, how much the deviation of the head is taking in account to the location. I mean, because if you move your head, you compensate the location. Or one way is, instead of locating the loudspeakers from one side to another side, is put the sound in the center and put on the sound in the center location of the spatial distribution. It would be a different, let's say, test. Mm-hmm. Do you know what I mean? Because if you give one sound from this side only, if your head is moving a little bit like that, you're locating more in this side. Right. If you put a sound in front and have the azimuth of the sound very in the center of the brain, but you put the information in a stereo field, you can have tests differently. It's much more like the people do in binaural testing, with the headphones. So that means the two questions I'd like to ask you.
Okay, so with respect to the head movements, you're absolutely right. Moving your head helps you localize sounds very well. So these are brief sounds, so they're hard to localize. The monkeys have a head post on their head, and they don't move at all. Their skull is fixed, okay? So their ear canals are fixed, so that's not an issue, right? We have to do that so we can stick the electrode in and it doesn't poke too far, right? The people were in a bite bar, so they had the same thing, right, when I did that. But you're absolutely right. Moving your head is really going to muck that up. The first question was on cognitive load and, okay, so, and timbre. And these experiments that I told you about today, it's all 200 millisecond broadband noise, and you're absolutely right. To be able to recognize words, et cetera, it helps to have a model in your head. And what happens in aging is, and we do this all the time, what happens in aging is you start making up for missed words, right? So you don't quite understand what they said. Did they say fight or flight, right? They must have said flight because they're talking about this thing. And they will make up this model of what the conversation is about, right? That's based on all these filling in errors, right? So we all fill in errors, but it's usually just one word every now and then. If you're doing, 30 or 40% of what you're hearing is you're assuming is what you're thinking about, then you find yourself not understanding a damn thing, right? Because you're talking about mowing the lawn and they're talking about going swimming later or something like that, right? So what happens in what humans do to compensate usually is exactly what you said. They use what they expect to hear, and words are much more complex and easier to discriminate than broadband noise from a place. We're doing a bunch of experiments now on both young and old animals where we use amplitude modulated noise as one of the stimuli, right? And it's in the flutterish range, so they go from maybe 17 to 50 or 60 hertz or something like that. And what we find there is that the old monkeys are just having a heck of a time learning this task, right? So they're having a really hard time picking up, so they have to discriminate either the location, which they do pretty well, or they have to discriminate the frequency of the AM rate, right? So usually it goes faster or slower, and they really suffer from that. And humans do much better, and I think we do better because we have some other kind of context to put that in, right? But as far as I know, that's the extent of the thing. And what we've also noticed about cognitive load is in the young monkeys, they're very good at hearing, here's the first stimulus, here's the second, is the second faster or slower than the first? The old monkeys do way better if you give them two to compare to. It takes them 2 tries to listen before they can hold that in memory long enough to decide if the next one was faster or slower, right? So that's all I can say about that.
So thanks for the talk. You have linked this cortical plasticity now at the very end with the change of seasons. But what if I want to remember next spring why I learned this spring? I mean, is it? If this plasticity is changing the map, would it be interesting that there is like a possibility that contextually a cortical tissue can have different information or functions, and that it's not only going from one to another, it's not always forgetting one or overwriting one by the other, but just a possibility of, I don't know if you have considered this.
That's a great question, and I've thought of that too. It's what is known is that if you learn something, you learn a new task, and then you stop doing it for a while, you get worse, but you get back to as good as you were much faster than if you'd never learned it in the first place. That's pretty common. And so by this line of thinking, what I would predict, I guess, is so imagine that I took one of those monkeys that got really good and had all those map changes, and then I just put him back in his cage and gave him biscuits like the rest for 3 or 4 or 5 months, and came back. I predicted it would look almost normal, right? And I also predict that if I didn't put him in the apparatus again, he'd get fast, be good again, much quicker than a naive animal, and his maps would change back, right? So I think that what happens is, here's your neuron here, and you've got your synaptic way, it's all figured out, and you're just a normal monkey neuron, right? Something happens, and now you're needed to do a new job, right? And so you release some weights and you increase others, right? But you remember those ones before, right? And you strengthen them, and because you got all those proteins and all that scaffolding and all that stuff already there, right? You're just not listening to them so much. And now you basically have twin states that you can survive in, right? And the real cool question is, well, how many states can you have, right? I mean, how many different maps can be in the same, can this one neuron participate in over the course of the lifetime, right? And back and forth.
Would you agree that this is now only a very hypothesis, a speculation, that there is no research?
It's just a hypothesis. I don't know of any evidence to support or refute it. Yeah.
Barbara has the urgent need to jump the queue. So
Sorry, yeah.
stop her?
Yeah, I mean, in support of that, the two-state thing, the one really cool allometric fact about birds, seasonal versus migratory birds. So you think that migratory birds, they do this complicated thing of traveling. It turns out those are the ones with the small brains. It's the ones that stay in place and have to do summer and winter both that have the really big brains across birds. So one thing you can do is be the animal with a lot more brain, and maybe that's where you put your extra representation.
So going back to the age-related hearing loss, you suggested that this was may involve the cortical inhibition or loss of cortical inhibition.
Yes.
And that would seem to make sense then of your reverberation, because the loss of the cortical inhibition would then lead to changes in the dynamics. So initially the response could become more exaggerated than it would be in a younger brain, and then maybe the inhibition comes in later, or there's less of it, but it dampens down, and then there's some reverberation as a consequence of that. So I think there is quite a lot of literature showing how these interneurons are involved in creating smooth dynamics. So if you mess around with them, that's the kind of thing you might expect to happen.
The reverberations.
Yeah.
Yeah.
And then I also wondered, you could work with Steve on this, whether it's the microglia that are eating all the cortical interneurons.
They have tangles and plaques and all that stuff in their brains, so they're losing real estate as they age.
Sorry, only to add in this comment, because reverberation is the sonic parameter that gives cues about the space.
Right.
So that means where you are, it's much more concerned with the reverberation than with location. So if you are here, it's because even you see the sound where they are, but the reverberation gives a cue of the space. I guess reverberation is very important to these parameters.
Yeah.
So thank you.
When the light is on, it's on.
Okay, so thank you for the great talk. My question is, what is the role of reward on plasticity in the maps that you showed, like in the somatosensory maps?
Right. That's a really good question. That so when you're working with monkeys, reward's really tricky because they don't care about your career at all. They're completely indifferent about it, frankly. And so you have to give them something that they want to make that work. And some monkeys just don't really care about anything at all, and so they don't ever do very well, right? So what you'd like to be able to do is get the monkey to actually work and tell you what it really perceives and manipulate reward in a way, in a parametric way, right? It's very difficult to do. I mean, you can do it to people a little bit, but then you can't do the single unit recording and all that stuff.
But you had in your experiment like two different cues, right? Like in the first experiment, well, you had like the auditory frequency that was like changing.
Yes.
And then you had the tactile frequency that was changing.
Yes.
But then the monkeys were trying to pay attention only to one of the cues because that was the one rewarded, right? And in that sense, then you saw like changes in the cortical maps and the tuning curves only in the modality that was rewarded, right?
Right.
So in this sense, you could argue that actually there is a strong link between the modality that is rewarded and the plasticity that is triggered. So then I was wondering, like, what is the mechanism? If you have any speculation, what would be the mechanism that actually triggers plasticity in the cortex due to a reward signal?
Okay, due to the reward signal. So Well, if I speculated the last 15 minutes, so why stop now, right? Dopamine is going to, so we know, it's got to be dopamine, right? So Mike Kilgard's work shows that if you stimulate nucleus basalis, you get plasticity without attention or training or any of that kind of stuff, right? And Sharon Giuliano showed years ago that if you block acetylcholine, you don't get plasticity after digit amputation. So acetylcholine is critical for it to work, right? And norepinephrine has been shown, that's classic work from Olds and those folks, showed that norepinephrine was critical for plasticity based from classical conditioning, right? And dopamine, I don't follow this literature so much, but I can't imagine somebody like... has he looked at dopamine? Yeah, okay. Yeah, so yeah, all three of those guys are doing something. And, you know, you could say that, you could interpret that data to say that it's entirely based on the reward contingency and the dopamine that gets released. You could say it's entirely based on I was paying attention and now acetylcholine is there. Okay, you can base it all, probably whatever norepinephrine does naturally, right? You could blame it on that. So it's almost certainly all those things going on at once. And like I said, if you were to lesion the nucleus accumbens, so you don't have this dopamine reward anymore, monkey's probably not going to do the task, right? So you're going to be stuck with that. Right.
Yeah. I have a question about the protocol. Would you say that you would have the same result in a 3D environment with the speakers above the monkey? And also to come back to this idea of reverberation, to get the distance of the sound. So you have the orientation, the direction, but also the distance.
Yeah, so if you were to do elevation, I think... So if you were to do this experiment in elevation, I predict that CL would also have sharper, two-dimensional, I guess, receptive fields, right? And we had done studies before that showed that, but we could just do frontal space because, you know, 16 speakers in this plane is great, but how many do you need to do the sphere, right? Which is now suddenly impractical. So I would predict that in CL you're going to have sharper spatial tuning. It's going to be elongated vertically because we don't localize sounds vertically as well as we do horizontally, and it would match up using the exact same kind of modeling parameters, et cetera, right? Distance is really tricky, and reverberation. So this is in a sound booth with the foam and all that stuff. So it's an anechoic chamber, right? So there's not any of these reverberations. There's a lot of interest now in studying that. I mean, it used to be you had to have this perfectly bizarre environment that had no echoes and all these things in order to study hearing, and spend a whole ton of money on trying to get that to happen, whereas we're never in anechoic environments, right? Maybe in a forest after snowfall or something, but usually there's reverberations all the time, and we simply don't study it, right? But I would imagine that if I could have done this experiment in a regular room full of bunch of stuff that the monkey is comfortable being in, everything would line up much better, right? Because we're just looking at, I'm just counting up the spikes in this kind of weird stimulus that's not something that the animal is used to hearing, right? And trying to make some sense out of it as opposed to something that's like a real stimulus that the animal really does hear and listen to and try to deal with, right? So the ideal experiment to me would be to have the monkey in the woods, right? And have my 10,000 electrodes in its auditory cortex and have little microphones at its tympanic membrane, right? And what are you hearing and what is your brain doing? And it would probably make a heck of a lot more sense than taking a monkey, sticking him in a chair, making him look right this direction. My nose itches, I can't reach it, and I listen to these sounds that mean nothing to me except I want a little bit of juice, right? And my brain is going to somehow help me do that, right? So
Greg, in some of what you're showing is that these motor areas or the auditory cortex is sort of being very much driven by the statistics of the input it's exposed to, right? But you might want to put some constraints on that, otherwise you're completely at the mercy of what your environment is giving you, how your environment is addressing the system. So what kind of constraints do you see are playing a role in assuring that you keep on, let's say, exploring novelty in that input space.
Right. So why don't you just perseverate on one thing and change your brain into now all I can do is this one thing? Right. Okay, so again, this is a totally artificial situation. I take the monkey, I say, you have to do this for 3 hours a day or you don't eat, right? And I'm actually surprised that, so what would happen if you said, okay, you have to do this or you won't eat, and then when you go back to your cage, it's not just a cage with bars, it's got all kinds of stuff that you want to manipulate, so you use your hands for other stuff too, then you're going to have to balance out somehow, right? So as long as you, moderation is key, Paul, I think. Otherwise...
I'll take that to heart, Greg. Thank you. Those are wise words to end up with. Thank you.
Thank you.
All right, it's lunchtime. We might have some practical messages here.
I don't know if they're practical, but...
Unpractical messages.
Well, just remember tonight is the beach party. And hopefully it's not raining, but bring a jacket and an umbrella if you're in a towel and your bathing suit. It's at 7 o'clock, so plenty of time to go swimming before dinner. Yeah?
Usually we try to play some volleyball, or football, or whatever happens.
Yeah, this year is left to the people. We always provide all sorts of things, but students don't like almost to swim, to touch the sand, so try very hard.
They're socializing,
Try
like,
very
on
hard.
the beach, having a great time.
Try very hard to like the beach, okay? And then for this weekend, don't go away. I know you're going to work very hard on your projects that have been assigned to all of you. So you all know your projects, right? Say yes, please. Yes. If there is anyone willing to work during the weekend, please. Well, both. Willing and wishing and hoping. And so please talk with the supervisor of the group, because then you will have to be signed up and to some list that allows you to enter the campus during the weekend. Okay? Is that clear?
And of course, we all encourage you to do that, because you all want to win the song competition, but you have to practice. Also over the
Don't
weekend.
confuse them, because they're all asking me, terrorized by you. Is it tonight? Is it tonight? It's not tonight.
Tonight is warming up. The real competition is 1 week from now. All right, enjoy your lunch. See you later.
Sisplau, tingueu cura de les vostres pertinences i porteu sempre amb vosaltres els objectes de valor. Por favor, vigile sus pertenencias y lleve siempre consigo los objetos de valor. Please, look after your belongings and keep valuables with you at all times.